techniques. Hello, good morning everyone. My name is Mahesha Poojary and welcome to the day 4 of the Gian program, Next Generation Sustainable Alternative Proteins for Food, Fundamentals and Technological Innovations. And today we are going to have 3 lectures. Lecture 7, which is about functional properties of food proteins, the classical functional properties, and lecture 8 is about how we can use alternative proteins to create meat analogs or dairy alternatives, and lecture 9 is about to create these meat analogs, how can we texturize plant proteins or alternative proteins, what kind of technologies we have, and I will be mainly talking about high moisture extrusion and, if time permits, shear cell technology. All right. So let's move on to the first lecture, that is lecture number 7 for today. Functional properties of proteins, solubility, water binding capacity, emulsification and foaming, and I hope I will complete it in 1 hour, but it is not always the case, and I will try to teach you as slowly as possible so you understand the basic concepts of these aspects, functionality and so on. That is the most important thing for me. I really do not want to show you the big tables and showing different functional properties of different proteins that you will always find it in literature. Google it, it is already there. But the most important thing for me is that you get the fundamental concepts. Make sense? All right, so that comes with my very first question for today. So yesterday we talked a lot about analytics, right? We talked about how do we and why we need to characterize plant proteins or alternative proteins, and what are the different techniques we have to characterize plant proteins or alternative proteins, right? So if you were to choose 2 different techniques, which one will be your favorite ones that you would choose or you would like to learn in the future? There are a lot of technologies I have mentioned. So now you get an opportunity. For example, you go to another laboratory for your PhD exchange and there is a lot of facilities. Which technique, at least one of them, that you would like to learn and why?
Hello, good morning, sir. Sir, I would like to love SDS page with chromatographic techniques.
Okay,
combination
why?
of these 2.
Why?
Because they will give you the exact idea about proteins, bands, and minor subunits of the proteins as compared to others like FTR and all this. So I would like to love these SDS and chromatography like LCMS to get the exact idea of which protein subunit is present in the sample.
Super. So he talks about STS gives him a better understanding of different bands and different bands related to different types of protein present in the complex food matrix here or a plant matrix here, and he can also complement STS page with, let's say, chromatography, and he can use different techniques to understand what kind of subunits present on a protein and how can we use mass spectrometry to elucidate the overall protein structure, and that is something he would like to learn. That is fantastic. So yes, anybody would like to mention something else that you would like to learn? Anybody? You do not have to be silent, it is okay. My class is a very interactive one and I like if you talk. Any other techniques did you like? Yeah, there is one person. There is nothing right and wrong, is please note that, do not hesitate, just take the mic and just talk.
Sir, first of all, good morning. I would like to learn fluorescence spectroscopy. I found it very interesting yesterday because it's first of all non-destructive and because of its ultra sensitive detection techniques, like we can really get
a lot
very
of
low concentration. If we have very low concentration, then also it can detect it.
Yeah,
So
yeah.
that's
I
right.
think that's an excellent point. Probably I also missed to mention that what she's telling that fluorescence spectroscopy is one of the interesting tool to learn because it is a non-destructive technology. That means you will get your sample back. So because sometimes your samples are so precious, you do purification and you get only in milligrams level and you want to do 10 different analyses with that particular protein, it's always nice to get your sample back. And fluorescence spectroscopy is one such techniques. You don't have much sample preparation, right? You just dissolve your sample in water or whatever buffer you say, take it to the machine, press button, excite and emit, and then you get a lot of data. Things actually starts in fluorescence spectroscopy after getting data, because then you can play around with the convolution, parafag and a lot of modeling, and then you can get more out of this fluorescence spectroscopy. And she's also mentioning that it's one of the most sensitive techniques. So small amount of sample is enough, because the excitation of an analyte is very efficient and emission is very specific. So you combine this selectivity, and that gives you very specific data about one particular molecule. However, we also talked about disadvantages. That is a nice technique, but you have to run it very carefully, because food is a very complex matrix. If you have unwanted things lying around, right, what we call interferences in analytical chemistry, that might compromise your fluorescence data. That you have to keep in mind. Thank you so much, and that's nice. And today we move on to the next important topic, that is functionality. And before going to functionality, I would like to show you what we have been studying so far. So we started on day 1, right? This is day 1, and we started about greenhouse gas emission, right? Right? We started with the cow. Remember my first slide? And then we thought, okay, because of this greenhouse gas emission, etc., sustainability and so on, we need to have a transition from animal based to plant based, right? Then on the second day we talked about a lot about, okay, how do we get proteins out, right? Different extraction technology we got, right? What is the product of this one? The protein powder, right? Then we talked about, okay, we have a lot of proteins, and how do we analyze, right? Analytical technique. What are the different techniques we have, right? Now we are getting a comprehensive understanding of the plant proteins or alternative proteins, right? We understood why we need to do this one, and how we are going to do this one, and then what has to be done to better understand the structure, right? Yesterday we talked more about structure and functionality. Structure and composition yesterday. And once you know the structure and composition, I've been telling structure is a very important thing, because that determines the day 4 what we have been studying today, F, the functionality, right? They're all interlinked, right? Please note that food is a very complex system. At the end of the day, we need functionality, right? Somebody has to eat it, right? And eating is a complex phenomenon. Everybody is unique, everybody has a different tongue, right? And that makes that we need to create different varieties of food. As I said, imagine dal and rice is very sustainable, imagine, but you can't serve dal and rice to the entire population. Just to give an example, we want yogurt, we want meat, we want many things. When you need this many things, we need to create new proteins with new functionalities or a better functionality, or something, the functionality that is equivalent to what we have today. For example, one of the best functional food, let's say milk, right? It's a colloidal solution, right? The beauty of milk, you see that it's very stable solution. Do you agree? You go to supermarket and take the milk can and see it. It never sediments unless milk is spoiled. Unless you store it for like several months after UHT treatment, it doesn't sediment. It's still a very stable suspension. Do you agree? It's very stable. Now if you take meat as an example, it still has a particular texture, right? You take a yogurt as an example, it has that creaminess, right? You take a cappuccino, the coffee that you buy, the fancy one, right? It has like nice foam on the top. So we need all these different things in food so that we can satisfy human beings, so that consumers can buy them, so that we can have a business. When consumers buy, then there is a transition from animals to plants. So what I want to say here is that it's important to create different functionality to satisfy the consumers, to have a transition from more animal based to plant based or alternative. Does that make sense? Why we need to do get the functionality? All right. So now I can ask you a question. What do you mean by a functionality as a scientist, as a food scientist? What do you mean by a protein having a functionality? You have heard this word, functional properties, functionalities, techno-functional properties. All these words you must have heard, right? Or read in the literature, or you may be writing your own research article on this one. What do you exactly mean by functionality? Yeah.
Water holding capacity oil holding capacity, foaming capacity, and all that it gives the texture to the protein.
So she is telling that there is different properties and all thus the giving the different texture to the protein or food in general, because food contains protein, right? Texture, she is telling about functionality, something about texture. Is there anything you can add? Is it only about texture? Yeah, she has a point there.
Properties of the certain chemicals can
In the proteins, so she is thinking that functionality is nothing but it's some sort of properties. Okay, maybe that properties again, as she mentioned, water holding and foaming, emulsifying and blah blah blah, many things are there. Anything else? What do you mean by functionality? One second, please take the mic behind. Yeah.
Characteristics of proteins that are desirable to make any desired product for the customer.
So he is thinking about it something kind of again properties, but this is not a normal property, it's a desirable properties, right? An important property that is important so that we can produce food products that consumers can buy or we can eat. But it's basically creating, it's basically creating a building blocks in my world, right? At the end of the day, texture is one of the properties, right? In addition to texture, we need juiciness, right? In addition to juiciness, sometimes we also need some sort of flavors, right? In addition to this one, we also perhaps need other properties, what would we call water binding properties, synergies and so many other things. So all of them at the end of the day create a building block for food matrix, and that can comprehensively termed as techno-functional properties, okay? And there's a hell lot of technological functional properties have been studied, and if you just browse in the internet functional properties of plant proteins, you get tons of them. And whenever a person isolates or extracts a plant protein for any other proteins, he or she will start analyzing as many as functional properties as possible, right? With the idea that, okay, if to find out my protein, what it has in terms of creating new building block for the proteins, for the food. Make sense? So it's very complex. There are a lot of different functional properties. The end goal is to create building block for the food, that again, as he mentions, for the consumers. Now, I keep telling that functional properties related to the structure of proteins, right? It's not only the structure, but people also talk about other things also nowadays, but mainly the structure determines the functionality, and because the structure is responsible for the behavior of protein in solution and in solid state or in semi-solid state, right? Some foods are solid, some foods are semi-solid, some foods are liquids, right? So the structure of the protein that determines their behavior in different phases, right? Because the structure is nothing but different amino acid composition. We have a primary structure, we have a secondary structure, we have a tertiary structure, we have a quaternary structure, and many of these levels of protein structure determines the functionality, right? So that keep in mind. And the structure is also responsible for interaction of protein with other molecules present in food system. Yesterday I told you, protein is never alone in a food system, and nobody gonna use or can use ultra pure single plant protein in a food product. It's very difficult. I don't say can't use it. It's very difficult to get a single ultra pure plant proteins. Not possible at the moment, because that requires hell lot of processing, a lot of purification. As I mentioned yesterday, if you increase the purification, you get the low and low yield. So you can't supply the demand. So that's the problem. So remember that the alternative protein worlds today is a complex mixture of different proteins, and different proteins are associated with different components. For example, they add binding agent, they add other emulsifier, they add antioxidants, they add carbohydrates, they add fat, they add water, and then everything is mixed, everything is processed. So too many things are happening in the system. So remember one thing in your mind now when I start functionality, alternative protein is not a single protein, it's a mixture of too many proteins. Okay, whenever we say functionality in plant world or alternative protein world, it's not associated with single protein often. it is associated with multiple proteins. For example, you are determining the pI of your P protein isolate, right? You get a pI diagram like this, right? It's pH, and this is solubility, and you say that okay, pI of P protein is, let us say 5.0, just to give an example. But when I say pI 5.0, it is not the pI of one particular protein in P protein isolate. It is a generic pI of all the proteins that is present in that particular solution that gives the lowest solubility. Does that make sense? The story is different for the milk proteins, because it is very possible to isolate individual milk proteins, let us say alpha lactalbumin, beta lactoglobulin, and bovine serum albumin, alpha casein, beta casein, kappa casein, whatever. We can get it as pure as possible in an individual form, but that is not the case for plant based proteins. So, whenever you read about functionality, keep in mind that it is never an individual protein, it is a mixture. Am I clear? So, whenever I talk in the next couple of slides now, do not attribute the functionality to the one protein, think about it as a complex mixture. Okay? Now, the protein, as I said, it is never stays in alone in the solution, and the functionality is because of structure. I am just repeating what I have taught to you last 3 days. The protein is made up of again amino acids, and each amino acid has its own properties, right? Some are polar, some are non-polar, some are having a charge, some are having a negative charge, right? Some are having the benzene ring, okay? Some are having the benzene ring with hydroxyl group, some are having the indole ring, and these molecules are very diverse. Amino acids are very diverse. That means there is a different reaction sites on the protein. Can you see? Different interaction site or the different reaction sites on the protein, and thus they can interact differently with other proteins. When I say protein interaction, it is not only protein interaction with, let us say, carbohydrate. No, protein can interact with other proteins. Protein can interact with polyphenols, right? Everything is attributed to the structure of proteins, individual amino acids. In addition to that, the proteins are folded, 3-dimensional structure, tertiary structure, and that is maybe responsible by hydrophobic interaction, hydrogen bonding, and disulfide bonds. Can you see? Now, when you start processing the food, you will start changing the structure of food, right? Proteins can be folded, proteins can be unfolded, right? When they fold and then they unfold, the availability of the sites for the interaction or the reaction will change, right? In this size, for example, tryptophan is buried, it cannot interact. When you heat, maybe it denatures, now more protein amino acids are available, maybe it is interacting differently, right? And that is also meaning that it interacts with different components with different ways depending on the 3-dimensional structure of the protein, then the functional properties can also vary. What I want to say here is, when functionality is because of structure, when the structure changes even to a minor level, functionality can be changed, and that is what we do in food processing. Do you agree? We want to change the structure of processing, the structure of proteins. We do ultrasound treatment sometime, we do cooking sometime, we do boiling sometime, we do pasteurization sometime, just to change the protein structure, and that can give you different functionality by interacting with different components in that complex system. Is it clear? Is it boring? No? Is it clear? Perfect. Now, I will just move on to, I have explained that slide already. Now, I will just move on to the important functional properties. The first one is protein water interaction. The first one is protein water interaction. Now, when you create a pea protein isolate or soy protein isolate or even milk protein isolate, whatever the proteins, right? It's very common that you would like to check how it behaves in water. Do you agree? It's a very fundamental thing we do in food science, because almost every food contains water to a lesser or greater extent. Do you agree? Milk contains 80-85% water, fish contains water, chicken contains water. If some foods contains low amount of water, perhaps the dried food and so on, but again you will cook it, right. Again, at some point of time you will definitely mix food with water. If you do not do any time, at the end you want to eat the food, right, you will mix with water in your mouth, right. So, then you will mix with water in your stomach. So, food proteins always come in contact with water. If not in the food, it will be in your mouth. So, the protein water interaction is an extremely important phenomenon we need to understand the protein functional properties, because it is the most common interaction that happens in food system, protein water interaction. Now, let us little bit dig into what do you mean by protein water interaction. I know you already know all these things, but I will try to cover some fundamentals only just to give you a better idea. Is it okay? Now, I have a glass of water, I take a pea protein isolate and I put it on this one. I did not shake it. I have a glass of water, I took a scoop of pea protein isolate and put it over the glass. It might look something like that. Can you see? It is a picture from the internet. Can you see? Does it dissolve quickly? No, right. What do you see actually often? You see turbidity after some time, if you shake it, right. But often what happens with plant proteins? They precipitate only after some time. If you shake it and then they precipitate, but immediately what you see? It just intermediate position, just stays on the top flowing, right. Have you seen that one? It is very normal. You take any, but it is not the case with sodium chloride, right. You take sugar or salt, you put it into water, it does not stay on the top, does it? It just goes down and then immediately start dissolving. Even if you do not shake it, it is try to dissolve somehow because of change in the entropy of the system, right. But proteins are not like that, maybe because they are big in size, right. Maybe because their particle size are so big compared to the particle size of other things. Maybe because they are not interacting with water as much as compared to sodium chloride or sugar, right. What I want to clearly say here is that proteins are different. They do not like water as such compared to other molecules, right. You need to do something. So, as soon as you add protein on a water, the first thing happens is what we call wettability. That means you have a water and air interface, right. The top layer is water, then you have an air, right. Now, the protein stays in that particular interface and try to wet itself, wettability, and it is an important property. That is the first stage of interaction between protein and water molecule, right. And then you will slowly swirl it, imagine, not like this one, you slowly swirl it. What happens? You start dispersing, not dissolving necessarily, right. It starts slowly going to different part of the solution and start moving inside the solution, right. So, that is what we call dispersion. Basically what happens during dispersion is you have a larger particles of proteins, right, and now it is coming in contact with water molecules slowly and the particle size is reducing, right. That is what is happening during the dispersion. And then if you are still continuously shaking, what would happen? Dissolution. Now, that is what we call solubility, right. It is a 3-step, it is a lot of different steps. So, the first step you had a water molecule, the protein was staying on the top, wettability, and the second step it start coming inside, but it is not soluble yet, dispersed on the solution, particle size are reducing, and in the third step you will see dissolution. That means now water molecules will start interacting with proteins more rapidly, right. Solubility, the protein will be solubilized, right. Now, if there is a more protein than the water can dissolve, what happens? Precipitates. That is the end stage, right, sedimentation. So, these are the 4 basic things happens when you take a scoop of alternative proteins and put it onto water. It is rare, in the rarest case you will get completely soluble clear solution. Do you agree with me? Of course, if you use extremely low concentration, it might get a very clear solution. I do not say no, but I am talking about the scoop of plant proteins in a glass of water, you will always see this phenomenon. Am I clear? Wettability, dispersibility, solubility and precipitation. These are the 4 generic protein water interaction we can see as soon as you put pea protein isolate, whatever isolate on water molecule into a glass of water. Now, I'll ask you a question that why this is important in alternative proteins world. Can you think about why do we need all this solubility information? Why do we need all this protein water interaction? Yeah.
Because solubility is required for all other functional properties. It is prerequisite for other, suppose foaming, emulsification, whatever the application is.
Yeah.
So solubility is required for every.
So what he is telling that the solubility or protein water interaction is the fundamental thing that determines the other functional properties of protein, right? It has to interact with water molecule, and then perhaps it will change the emulsification property and maybe gelation properties, maybe whatever properties you are saying. It's something related to solubility. If the protein is completely insoluble, dead zero solubility, then probably that is not a good protein to be used in food system. You can use it to make a cardboard or something. I don't say there is no application. You can make it for the packaging material, for example. Protein is not only for food, it can be used for some other purposes. So you can find other application. For food purposes, you need to find to a certain degree of solubility. I don't say it has to be 100% soluble. If it's 100% soluble, probably it's very good to use it in sports beverages, for example, infant formula, for example, where it's very important to have kind of a solubility. Okay? Any other reason? Think about texture. What's that? Juiciness, right? If you go to KFC, we eat chicken there, and one of the things we like that crispiness. In addition to that, we also like the juiciness, right? It's a fine balance, right? And if chicken is very rubbery and dry, we don't like it. I often buy biryani somewhere in the hotels, right, in the restaurants, and if the biryani, I get this so-called breast piece and what we call, it's so hard sometimes because of it has lost all its moisture. I really don't like it to eat because I don't see that juiciness, right. Sorry, I'm talking about plant-based, now I'm talking about chicken biryani, don't mind. In the future, biryani chicken will be replaced by plant-based chicken, don't worry about that. So, but we still want to get that juiciness, right, and that juiciness is something related to protein water interaction. Let us take this few examples here. It is also important to create plant-based milk, right. Milk should be milky. If milk is like yogurt, do you buy it, right? If milk is a hard solid, will you buy it? It's a different food. So, we need different properties. So, yogurt has to be soft, chicken has to be juicy, milk has to be milky, all these properties coming from protein water interaction. There are, of course, there are other interaction, but one of the important one is protein water interaction. As I just mentioned, the solubility, this is again a high school thing or a primary school thing. We all studied this equation, right? Solute plus solvent is equal to solution, right? This is something I always by hearted when I was maybe in primary school. Solute plus solvent is equal to solution, because that's a common 1 marks question, right? So, solute is nothing but the analyte, in this case protein. Solvent could be water, and solution is nothing but it's a homogeneous mixture. It's a homogeneous. When you say a solution, we have a homogeneous solution and heterogeneous solution and so on. So is all good? Is everything okay? You want to go? Yeah, you're welcome to leave, no problem. Yeah. So if you want to go for some reason out of the class, you don't have to take my permission. Just go, and if you want to come back, you come back later. You don't have to take my permission for that. Just leave and come back. If you want to come back, you come back. Okay? so there is no question about that. We were talking about a solution where the solute dissolve in solvent becomes solution, right? Now, in case of protein, the solubility is dependent on the concentration at which homogeneous solution is formed. Okay? Homogeneous solution, you know, right? Everything is in one single phase. It's called homogeneous solution, and that is called solubility. If you have a precipitate, that is not solubility. If you have a dispersion, this is not solubility. If you have a colloid, that is not necessarily a solubility. The solubility is nothing but you have a clear solution, in other words, right? And the solubility takes place in a different phenomenon Let us try to explain that one. Now I have a beaker of water, right? I have a beaker of water, and this is me. Now I have a protein powder in my hand, all right, and then I will add protein powder to this one, okay. I had a very small quantity initially, very small quantity, okay, and the solution is continuously under stirring, okay. We are stirring it continuously. Imagine the protein I am adding ultra pure and having a very high solubility. I am adding only small amount and it is vigorously being stirred. What would happen? It will completely dissolve. So we are in this particular phase at particular time, right? We have completely soluble. Please note that solubility is again dependent on temperature, pH and other things, right, and also pressure. So atmospheric pressure, temperature, pH, these are the two governing factors that determines the solubility. Keep that in mind. I will get more details into that later. Now I did not stop here. I add one more scoop. What would happen? It might take time, but if a solvent can dissolve that solute, that is protein here, it can still solubilize. Do you agree? It can still completely solubilize, right. We are still in this particular region now. Now I will add another big scoop, little bit bigger scoop. What would happen? We are reaching the so called saturation point. Out of that big scoop, maybe half of them soluble, the remaining half is do not want to solubilize because solute is not interacting completely with the solvent molecule. Now we are reaching the so called saturation point, the metastable state, right. And what that time we come somewhere in this region where we are dissolving the proteins to a certain extent and the remaining proteins will start precipitating. It is not the direct precipitation, it is what we call the first step of that precipitation is something called formation of protein crystals or amorphous particles basically. What exactly happened here is that there is a situation where you have completely solubilized proteins and then you are adding more and more proteins now, right, and that protein we really wanted to solubilize, but it cannot, right. Then it stays in the solution as a small particle. If you add more and more particle, that will increase in its size, right. Its nucleus will increase the size. So it is getting a bigger and bigger particle. Sometimes we call it as aggregates, right. The proteins will size in the increasing in size, and if you still add more and more and more protein, if you increase the concentration further, what happens? Then you see complete precipitation of the protein, right. Do you agree? Supersaturation. You will get the complete precipitation of the protein. That is a system where we have two things, protein in precipitate and protein in solution. So that is where we are here. And this is how the protein solubility changes depending on the parameters. Parameters are also important. Okay? By changing the parameters, you can change this diagram. Do you agree? By changing the parameters, you can change in the paragraph. For example, some proteins need very alkaline condition to completely dissolve, right. Some protein might need mild heating. Some protein might need a little bit higher heating to completely solubilize. So by changing the parameters, you can change the solubility behavior and get the more and more protein into solution. But no matter what, at some point you cannot solubilize, things will precipitate. Okay, and that depends upon the concentration of the protein you are adding and the temperature, pH and other parameters and ionic strength and so on. Any question until here? It is very, very fundamental thing I am teaching you now. It is not something new, it is very simple one. How to determine the parameters for the solubility. So how to determine, okay, which protein can solubilize better than this one? I'll explain that in the coming slide. There are some slides about that. Good question. Okay. Yeah.
Sir, generally when we test the solubility, we don't just say it at the right concentration. We generally prepare a solution of 10 mg per ml or something and we report it. That's how everybody
Yes,
does.
yes.
So how does it make sense in real life situations?
Absolutely good question. I have one complete slide on that one. I'll get back to you that question.
One more question, sir.
Yes, yes.
So when I dissolve my protein initially, it will not be soluble. But
No.
if I hydrate it overnight,
Yes.
the next day it becomes soluble.
Yes.
So how does it affect? So can I say that after hydration of 24 hours, at this concentration, my protein is
Yes.
completely soluble?
Yes, that is all. Yeah, I will also cover that point also. It's all the problems of the plant-based people, right? I'll try to cover all these points. If I don't cover, remind me again. Okay, there are some points about that. Yeah, good questions. All right. So coming back to your first question, how are we measuring solubility today in the alternatives world, right? We are measuring it by 3 different methods. The first one is relative solubility, the second one is maximum solubility, third one is predictive solubility. The first one is the question you asked, okay? The last one is the question you asked, right? We will try to cover some of these things, okay? These are the 3 things we are using at the moment to determine the extent of protein solubility. Let us begin with the relative solubility. How many of you have done solubility analysis in your master's or PhD or bachelor's, I don't know? Okay, we have 2. You guys never did, did you try to dissolve any protein in your life, other people? No? No, you must have made chai and you must have made chai with milk powder, right? In India, it's all very common. Yes or no? Does it contain protein? Yes, so you have dissolved protein, I'm pretty sure. You don't know. So, all right. So, now I am talking a little bit about scientific, how do we do it in a research paper to determine the solubility. Is it okay? Not making chai. So, the first one is that the relative solubility, okay. This is something most commonly you find it in the alternative research paper. What do you mean by alternative research paper? Research paper focused on alternative proteins, okay, not alternative. This is a wrong statement, okay. You will see that solubility analysis, they always 90% people use this method, relative solubility measurement. It's nothing but, it's not finding out the absolute maximum solubility of the given protein, just to find out the solubility of the protein you have extracted or you have purchased to in a particular condition. Am I clear? What is the solubility of a protein in a particular condition? That is relative solubility. We do not care about how much it will dissolve, how much more it will dissolve, how much less it is dissolved, we do not care about it, okay. At a given condition, what will be the solubility? That is relative solubility. And what are the given conditions? Do you remember? What do you do? What did you do in your own experiment? You have the mic, yeah, yeah.
I have dissolved 10 mg per ml of protein.
Yeah. So she is telling the protein concentration is approximately 10 mg per ml.
Yes.
Okay, it's 1%. Yeah. Next.
So after dissolving it,
Yeah.
I have adjusted the pH.
Yeah.
At what particular pH I have to?
So she adjusts the pH from X. I don't know how much pH she is exacting, but you will try to adjust the pH.
Yes,
Yeah,
I have
yeah.
adjusted it to 7.
You would adjust it to X is equal to 7 here. Yes.
So at particular after adjusting to pH 7,
Yeah.
then I have gone for the protein. Then I have centrifuged it. Then I have
Did
collected
you
the
wait for
supernatant.
some time or
Yeah,
no?
yeah, yes,
So
I
what?
have.
How long did you wait?
I have waited for 2 hours. I have
So she
kept it in
stirred it
stirrer.
for 2
2
hours. So time is now 2 hours. Then what did you do?
After that, I have centrifuged it.
So then she used centrifugation, another processing here. What? How long? How many G?
It is for 9000 xg.
So she is telling 9000 G.
9000
and
g at for 20 minutes.
for 20 minutes, right?
Yes.
And then
Then I have collected the supernatant.
you collected the supernatant. So you have a centrifuge tube and you have a protein pellet here and you have a supernatant. Yes, yes.
And I have gone for the protein content analysis of that supernatant.
What method did you use?
I have used Bradford method.
So she used Bradford method to determine protein content. Now can you see here what did she do? She used a particular conditions here, right? She used pH 7, she used time 2 hours for stirring, she used centrifugation conditions 9000 G for 20 minutes, she used Bradford assay. Now I'll ask her question, why did you choose these conditions? Who told you to choose condition? What is the rationale behind this one?
I have referred many papers, sir.
Yeah.
So based on that, I came to a common condition.
Exactly. Everybody is unique here. I mean, she read so many papers and she thought, oh, this is the good, many people are using this condition, let me also use, I'll get this one. And that's what I also do in my lab. right? I'm not telling it's wrong. It's a relative thing. Am I clear? That's the whole thing of relativity. We check everybody. Oh, this person is also doing that one. That person is also doing that one. Let me try to do this one. I don't care whether it's right or wrong.
Because I can compare it with someone.
Exactly. And this, I'm not telling it's wrong. Please don't take it in mind.
Yeah.
What I'm telling is not wrong. What you've done is not wrong. What I'm telling is that that's how this method is based on. It's very relative. On a particular condition, she's trying to understand the solubility. Now I'll ask you a question. If you change pH to 8, don't you get the different result?
Yes, I will get a
Exactly. If you change the pH to 5, don't you get the different
Yes,
result?
again
If you
I
make the centrifugation speed 6000, don't you get a different result? Instead of Bradford assay, if you use BC assay, do you get a different result? Instead of BC assay, you go for the Geldahl CHNS analysis, you get a different result. So depending on the method you are using, you get a different result, and that is also one of the disadvantages of this method. Am I clear? So the relative protein measurement is nothing but measuring the protein under a different and a given conditions. I'm just trying to show it in an illustrative way. We take the protein, we set the conditions, we centrifuge it, we measure the protein content, and the relative solubility is nothing but the ratio of the soluble fraction to the non-centrifuged whole fraction. That is the relative solubility. Okay. Advantage is easy to do, may be comparable with other people, but it's also a big problem. I've been reading papers on relative solubility. You can correct me if I'm wrong. It's not that everybody follow the same condition. You said 9000, I find 20,000 G here. I also found somebody centrifuging at 6,000 because their centrifugation machine does not go to 20,000 G. Then what they will do? They'll stop doing research? No, they will increase the time. So this is a game, right? And okay, some people want to adjust the pH, right? Every protein has a different buffering capacity, you know, right? What do you mean by buffering capacity? You take one scoop of pea protein isolate and shake it and measure the pH of the solution. It says 7. You take rapeseed protein and measure the pH of solution. It says 8. Soy protein may be saying some other pH. So every protein has create different pH in the solution. Then to adjust it to 7, what you will add? You add HCl, you add NaOH. Again, you are adding more and more salt, right? You know the salting in, salting out, and blah blah effect. Last time I taught you. Again, that will change. So it's not universal. Things will change depending on what you are doing. So it's not also readily comparable. And please note this point, and this is one of the challenges in comparing different research articles, comparing the properties of different plant protein isolates today. Nobody is using a homogeneous method. It's not readily comparable from paper to paper, and that's one of the challenges for food industries also. They go crazy. They have to do their own methods now. Make sense? So we need to find a uniform method that everybody follows to get the to do the comparison throughout the world based on if you want to compare different but it's not an easy task, just to let you know. We also covered disadvantages, and the first thing I already mentioned that it is based on specific conditions. Conditions vary. It's also depending on initial concentration. I forgot to mention that. What she mentioned that she used 10 milligram per ml. The question is that why are you using 10 milligram per ml? That's a very important point. Why? Because if I use a protein solution, it's 10 milligram per ml. Let's say it is 1% solution, right? I say my protein is 100% soluble. Do you agree? Imagine 1% is 100% soluble. If I use 1% solution, I say it's 100% soluble. Now my PhD student will come and he thinks, no, I don't want to do 1%, I will take 10% solution, right? Then what will be the percentage solubility? Is 10%. So suddenly we change the solubility of the paper, right? Now which one will I believe? She is selling the paper saying that my protein is 100% soluble, oh, it's super good. What is the concentration did you use? 1%. But the same your own PhD student, and he did another experiment, but instead of taking 1%, that person took 10%. Now the data will show it's only 10% soluble. So we are biased again. Am I clear? So the initial concentration also plays a role here. And now the hydration time, that's the experiment you mentioned here, is that you are stirring it continuously for 2 hours. Coming back to your own question, sometimes you do freeze drying, sometimes you do spray drying, and they have a very different properties and solubility. They need hydration time 2 days, 3 days, even sometimes 1 month also, trust me, to completely hydrate proteins. it's not a easy thing to understand. So the solubility varies because of all these properties. I'll come back on the drying thing in the next slide also. Is it clear? Any question? Will you use relative solubility in the future? Yes or no? If I say, I told you a lot of things about relative solubility, will you use or not? Yes.
So it is mentioned in the packet how many scoops need to be added and how much of the water.
Yes, absolutely. I mean, we still need that information for some reason, right? The only problem is you have to take the data very carefully from the internet. That's it. That's the whole conclusion. I don't say don't use it. You have to use relative solubility because 90% of the world is using the same method. So you go for it, but do it very carefully. Is it okay? Now I will come to, okay. Now I'll come back to the maximum solubility, okay. It's a real solubility according to me. What do you mean by maximum solubility is nothing but how much protein can really dissolve in a given solution, okay, not under a specific condition, okay. It's not under a specific condition, how much protein in a given volume of water or any other solvent, how much it can dissolve. That is called maximum solubility, and this is a very tedious experiment to do, almost impossible in more cases, right. Why? Because you do it in this way, you take protein powder and put it into water containing glass or beaker or whatever, falcon tube here, then you need to stir it forever, because the protein solubility is a very slow process, okay. Every half an hour, let us say 0.0001% protein is solubilizing, okay. Imagine that is the property of your protein, because protein has to hydrate, that means the water molecule should go and hydrate every individual moiety so to come into solution, and that takes a lot of time, right, and that can take even up to 2 months sometimes. I was reading an article, if you really want to determine the maximum solubility, you need to stir continuously for 2 months. Of course, I don't have 2 months for my PhD, right. So we don't do it in food science often, because that information is not necessarily needed in some cases. So just to tell you that if you really want to find out the real solubility, you need to give a good conditions to and find out the actual solubility, or slowly add more and more and more and more and more, stir and measure every time, okay, when do I see no concentration difference, and that will be the maximum solubility, and we don't do it often. Have you done it in your PhD? No. Make sense? So just to show you the maximum solubility curve here, don't read this text, it's just to, because I'll leave this slide so you understand this one. So this is the curve, basically you have protein concentration, there is no protein, no solubility. Increasing the protein concentration in this direction, solubility increases. Now the blue is pure protein. Imagine you have an ultra pure one protein, individual protein, okay, and the green one is isolate. Isolate is, let us say, mixture. It's always mixture of 10 different or 100 different proteins, okay, mixture of mixture. If you have an individual proteins, let us say ultra pure only one type of protein, then solubility increases continuously. At a particular point, it will stop solubilizing, because you are completely saturated, no more solute molecule is interacting with the solvent molecule, then you see the no solubility change. But in case of mixture, you never get this sharp thing, you will get this bent thing. Why? Because you have numerous proteins playing. I want to solubilize, I don't want to solubilize. I want to solubilize, I don't want to solubilize. So in that cases, you don't get a sharp change in the solubility. Just to show you don't get really like this beautiful curve. It's an illustration. Just to let you know that the mixture, you have a different solubility property compared to individual proteins. Okay. One of the disadvantages, it takes for a long time. And then coming back to the predictive solubility, you have been asking this question that how do we know which one we solubilizing more and which we don't solubilize more. Easiest thing is to do trial and error method. Take all the proteins, do solubility test, but nowadays we have something called AI, artificial intelligence and machine learning. Now, not for all the plant proteins, at least some of the plant protein sequences are available. You go to PDB and you find the protein structure, You have a complete amino acid sequences of some of the plant protein, not all, some of the plant protein. Milk you have, almost every individual milk proteins have been crystallized. You have a absolutely beautiful structure of milk proteins. Plants we do not have, but still we have some, and if you know the amino acid sequence, if it is in the database, they can create the 3-dimensional structure nicely, and they know which amino acid is on the surface, they know which amino acid is buried. If there is a more and more polar amino acids on the surface, what it will happen? It will solubilize more, it likes water more, right? If there is more and more non-polar amino acid on the surface, it will not solubilize more. You can determine these properties in the computer directly using machine learning, okay? And you can also determine the theoretical PI of the protein. What is PI? Isoelectric point, the lowest solubility. You can also find out the theoretical isoelectric point if you know the individual amino acid composition. You can calculate it theoretically. And when you know the surface properties, when you know the folding behavior, when you know the theoretical PI, you can predict the protein solubility. But it is not very easy in the plant-based world. Why? Because of the complex proteins, one of the reasons. The second reason is, it is not an individual one protein. You have 10 to 20 to 30, 40 different varieties in one protein isolate, and this computer also goes crazy, right? Because they are talking about individual proteins, but we do not have individual proteins there, we have a mixture of proteins. So, the property what you are measuring is a mixture of everything. So, to predict that is also very difficult. Am I clear? So, the easiest thing probably at the moment may be the trial and error, but if you develop this AI modeling, it is an important field nowadays. This is also something career you can take in the future, AI in food science, that can revolutionize things. Then people do not have to do lot of laboratory work. There is apparently a lot of models for that. I have no idea about what these models are. I just mentioning it here. If you are interested, go and Google about this. Okay, these are the models to predict the solubility. One of the disadvantages, as I mentioned, it is very complex system, then we cannot measure. Now, we will look little bit about the different factors affecting the solubility. Are you tired? Tired? Still have the energy? Okay. Now, we will be discussing about the solubility, and I will talk little bit about different factors affecting the solubility. The first one is temperature. Now, I have a beaker containing water, and now my protein is inside, completely soluble, imagine, or partially soluble, right? Now, I am slowly heating this beaker now, okay? What are the effects you will see? Do not read all these things written on the slide, just think about it. I have a beaker containing proteins, I am slowly heating it now. What are the effects you will see? Yeah, could you pass a mic on her? Yeah.
Protein denatures.
The first thing she was thinking that the protein might denature, because proteins are folded, right? And this folding is because of which bonds?
Peptide bonds.
Hydrogen bond. Peptide bond is also true, but hydrogen bonding. Peptide bonds you can't break by just heating, right? You need a strong acid. So, hydrogen bond can be easily broken, not very easily, depending on how many hydrogen bonds you have, can be easily broken, and proteins can denature, right? Every protein has its own denaturation temperature. Have you heard about it? Right? Some protein will completely denature at 80 degrees, some will be 50 degrees, some will be 90 degrees, some will be 100 degrees Celsius. For example, rapeseed protein has two proteins called napin and cruciferin. Napin denatures around 100 and 110 degrees Celsius, cruciferin denatures around 80 degrees Celsius, because they have a different temperature when they denature. So, one of the things you can observe, the protein can denature. Anything you observe when you are heating? What else could happen? Solubility will increase. Why do you think its solubility will increase?
When the protein unfolds, there will be a change in the bonding.
Yeah.
Like when it unfolds, the hydrophobic may come outside, or hydrophilic, whichever is buried, may come outside.
Yeah.
And it interacts more with the water. and so the solubility will increase.
I think I have a point here. Can you read this one? It's a very good suggestion. But I am writing that thermal denaturation decreases the protein solubility. What could be the reason? You are right. I am not telling you wrong. But sometimes the solubility is also decreasing.
Yes, because of aggregation it may decrease.
Exactly. Now these are mixed behavior effects. Can you see what she is telling? When you are heating, this was originally folded proteins, now it is denatured, right? And she is telling the water molecule can interact nicely and protein will solubilize. She is also telling this is not the case. When you heat it, it denatures and then it interacts with other proteins, it becomes a bigger particle and precipitates. Am I clear? So, it is very difficult to say what exactly is happening depending on the protein and the temperature you are applying and the heat you are applying. Make sense? Heat load. So, when you denature, protein can get more water molecules into excess, it might solubilize, but in many cases it not solubilizes. Why? Because interaction will be with water molecules may be less governed compared to protein-protein interaction, because you will expose hydrophobic regions quickly. Okay? You will expose the thiol groups, right? The protein will have so-called thiol groups like this, okay? And another protein will have a disulfide bonds like this one. And then you will favor the thiol disulfide bond linkages and you will get a protein with linked by disulfide bond, and that will give you the protein aggregation and the solubility can be compromised. It is a very difficult and important phenomenon to understand. That means you can't say the solubility is increasing. If you see increased, you are happy. If you see decreased, then you know the reason. Make sense? How temperature affects? Yeah.
Is up to 40 to 60, it may solubilize, but
Yes.
if you heat more than that, aggregation happens, and that's how we do gels.
Absolutely, yeah. It's the critical temperature, that is something you need to mind. Usually at the very low temperature, what happens, the hydrogen bonding making and breaking is more governed, right? At a very high temperature, denaturation is more favorable, aggregation will be more favorable, coagulation will be more favorable, so you see less solubility. Maybe, I don't know the curve reality, how will it look like, the curve might look like this one with temperature, right? You have increased temperature up to 40 degrees Celsius, then you see the decreased solubility when the temperature increases further. Make sense? Okay? So exactly what you are mentioning is that we used temperature to coagulate proteins to create gels. That is one of the methods, simply because you open up the structure, you create new bonds, because you allow protein to interact each other much better. All right, now let us move on to some examples here. So in solubility of Mung protein isolate, you can clearly see that at lower temperature also they have the decreasing solubility and no effect in the solubility. That is what the mixed effect. So it depends upon the type of protein, the solubility varies with respect to temperature. Now in this case they are actually doing this experiment where they actually took this Mung protein isolate and they did the ultrasound treatment along with heating. Then they saw kind of increased solubility. What could be the reason? Reduction in the particle size. So ultrasound, you know, remember ultrasound I taught you last time? You put this beaker containing protein and you put ultrasonic probe, use it for the extraction, but ultrasound is not only doing the extraction, it will also reduce the particle size. When you reduce the particle size, surface area increases, interaction with water molecule increases, solubility increases. But people also say that it will also improve the hydration of the molecule ultrasound because of the cavitation phenomenon. I am not very sure it is true or wrong, but they also say that it improves the hydration of water molecule by entrapping the water molecule on the surface. There are a lot of theories, but there is no proof for that one. But they can improve the solubility. Okay? pH. Can anyone explain? I am a little bit tired now. Can anyone explain how pH would affect solubility? Take a mic, just talk for 2 minutes. You are welcome to do that. How pH would affect solubility?
So this again we can go back to the isoelectric point of the pH.
Exactly. So we go back to our previous lecture. So we have pH, we have solubility. Can you explain me how would the curve look like?
The curve would look like this.
The reverse bell shape, right?
Yes.
So it can have a high solubility and solubility decreases, no solubility and then increases. This is what you see often, solubility. Now, could you please explain me why you see the highest solubility here? At lowest pH, you have the higher solubility. What could be the reason?
So
More? Yes, but why? Yeah, yeah.
At lower pH, protein will have more sites for bonding.
Yeah, more sites for bonding. What exactly that site?
The OH negative. And
OH negative?
they will interact with the water H positive.
Would that have a OH negative at acidic condition? I mean, you're coming to the right answer, but it's not the absolutely right answer. So he's talking something about the charge, right? Now, can you tell me, I have a protein, okay? This is my protein. The pH of the solution is 2.0. What will be the charge of the protein? Likely, yeah. Likely they will be positively charged. Do you agree? Why? Because the amino groups on the protein from lysine will be protonated. Arginines will be protonated. So, protein will get net positive charge. Do you agree? Positive charge is ionic. Water likes ionic compounds. So, protein solubilizes here. In this region, proteins will be positively charged. The positive charge decreases when you increase the pH. So, the solubility decreases. Here, you will find highest solubility. What could be the reason? More negative charge, because you will have more deprotonation, the carboxylic acid will become COO minus. So, the protein will have more negative charge is ionic, water likes ionic. So, solubility increases. What exactly happening here? No net charge, because there is a 50% positive, 50% negative. So, the protein have plus minus and so on, minus plus minus so on, equal charges attract each other, they precipitate off. Clear? Please do not forget this curve. Okay? It is an important curve, you have to keep it in mind. It is very useful to do the alkali extraction of your protein, right. This is exactly we do. We extract the proteins here, we precipitate the extracted protein here, right. We extract the proteins here, we precipitate the extracted proteins here, exactly based on this phenomenon. Am I clear? But I will explain a little bit beyond these things now, just think powerfully. Usually, alkali condition also denatures proteins. Okay. Why? Because when the protein is here, this 3-dimensionally folded, you have a disulfide bonds, right. It is known that because of alkali condition, because of this OH, strong OH minus ion, this disulfide bond can be broken. There is a lot of theories about that one. I have already told about and I will come back to that later. Just for now, you just remember that this disulfide bond can be broken because of beta elimination reaction, because of OH this one. Now, the protein becomes open like this. Can you see? You get an SH group or something like that. Now, the protein denatures, what happens? My previous, I was telling when the protein denatures, aggregation happens, right, because you will allow more and more protein aggregation. But if you have more and more protein aggregation, protein has to precipitate, but the protein do not precipitate here. Can you tell me why? Now, two phenomena are happening simultaneously. One, proteins are getting negative charge, meantime proteins are also denaturing, but still we see highest solubility. What could be the reason? Am I confusing you? Huh? In this, are you here? Can you follow me until here? What is happening here? I mentioned in this region, the protein will be having negative charge, okay, and the negative charge will help protein to solubilize. But in the same region, the protein can also denature. When the protein denatures, they tend to aggregate. That is what I told you in my previous slide. So, I also expect the protein to aggregate and precipitate, but we do not see any precipitation often here. What could be the reason? Because the charge is more than the aggregation. Why? Why there is no aggregation?
the alkali condition that we give
Yeah.
is is more and more when compared to the aggregation.
So, she is telling that the effect of getting negative charge is overshadowing the
Yes.
effect of denaturation.
Yes.
That is what your explanation. Okay, I fully agree with you. But actually, that is true. What exactly happens in this particular region? The protein gets negative charge. Other proteins, everything gets negative charge. To aggregate, two proteins would come together. Do you agree? When they aggregate, they come together, then they aggregate. Now, the electrostatic repulsion, now everything is having negative charge, they want to come together because they want to aggregate, but you have a negative charge on the surface. Ionic bonding is very, very strong, the repulsion is so strong, it pulls away, so they do not aggregate and precipitate. Does that make sense? So, I am talking about two different theories here, and which one is more governing than the other one. Okay? Could somebody summarize this one for me in 30 seconds, what I just explained? You try, you have a mic right with you. Why we see highest solubility and alkaline condition even though protein aggregation is more favorable there? That is the question.
At alkali pH, there is more negative charge. So, there is more electrostatic repulsion that does not allow it to settle.
Does not allow it to interact together
with each other and precipitate.
Settle. Is it clear? Is it too much for you? Yes or no? Are you getting or not? I am explaining too much, or you want to? Do you want me to explain less and move? You are welcome to give me the feedback. You are getting the point, right? Yes. Okay. All right. And you have any question? Yeah. Any question? No questions? Good. Let us move on. So, another important observation we can make in this particular figure. Can you carefully look into this figure? I will give you 30 seconds. Look into this figure and give me an observation. 30 seconds starts now. Do not worry about this content now, okay. Just to look into the figure and make an observation and tell me what is the observation. You can hope you can see the color properly, there is a gray and dark black, okay, lines. I hope it comes nicely in the screen. Yeah. Okay, 30 seconds is over. Do anybody have a point to make? Any observation you make here? Yes.
Sunflower globulin and pea globulin are not same.
She is telling that the globulins are not same. I agree. Any other thing? Any other thing did you observe? She has an observation.
Depending on the protein fraction, the solubility changes.
Depending on the protein fraction, solubility changes. I agree. But what exactly you are observing in terms of solubility? I give you one hint. Can you compare globulins and albumins here?
No, we cannot. We can compare sunflower globulin with pea globulin, whereas we cannot compare sunflower globulin with sunflower albumin.
I agree with you, but forget about that for now. Just compare globulin versus albumin in these cases. Just compare in one of the figures. Just compare globulin versus albumin. What do you see?
So globulins have a very predictive solubility, the trend which we expect.
Yeah.
But albumins have a very unpredictable behavior.
Can you see that one? The globulins have a beautiful curve, what I have just mentioned, like this one, right, in both the cases. Albumins, look at the solubility percentage. Can you see here? Who is soluble, more soluble? Who is soluble? Albumins. Can you see? Albumins are more soluble, that is one particular observation. Second observation, as he says, globulins have a very predictive solubility depending on the pH, albumins are crazy. And not only that, you do not have a very clear PI pattern. Can you see here? This is the PI we call most often in food science, but you do not see a very clear PI here. And even at a different pH, we are talking about 1 to 11 here, is soluble. How much? 80%. So, this phenomenon what I just explained over here, not applicable to every proteins. Okay. Please note that albumins having a greater solubility, even though they have a particular pI, they might have high solubility in their pI. Make sense? Albumins do have a pI, but it's don't believe a fact that every protein will have the lowest solubility at their pI. That is not true. Albumins can be soluble to a greater extent at their pI. Is that okay? Does that make sense to you? Because they don't have this clear predictive solubility curves. Am I clear? For example, nepin is a rapeseed protein. I think it has pI around 10 or something like that, if I am not wrong, and it's completely soluble in a very long pH range, albumin. Okay? And it's an important thing you have to keep it in mind when you do protein extraction. When you do protein extraction, you extract at alkali condition. Do you agree? You extract at pH 11, for example. Then you do isoelectric precipitation. You often use one pH. What is that? 4 or 5. Do you agree? You use 4 or 5 as an isoelectric pH, and then you precipitate the protein and you freeze dry it, and you say, oh, I got a lot of field. But actually, you are only extracting globulins. Can you see? Albumins are still in the supernatant. Why? Because albumins are still soluble at that particular pH. Does that make sense? So, when you do extraction study, be careful that don't believe you are precipitating everything. No, you are only precipitating the small portion. Often you lose a lot of albumins in isoelectric point precipitation if you work with the plant proteins. Supernatant still has proteins. That is our albumin. It is very important to mention. Why? Because it is a good thing in the plant based world, because most of the storage proteins are globulin, fortunately. We get a lot of proteins, we are very happy about it, but you still lose a lot of albumins. But albumins have a different functional property than globulin. So, if you really need albumins, you need to refractionate the supernatant. Make sense? Yeah.
Sir, when we do Osborne fractionation of our protein,
Yeah.
that time we dissolve our protein in we dissolve the raw material in water,
Yeah.
and then we precipitate
Yeah.
at the pH
Yeah.
to get albumins.
Yeah, in Osborne, yeah,
Yes.
provided they precipitate.
Yes.
But I mean, I have some, but if you look into plant-based proteins, I think some of them will precipitate. I agree with you, albumins, but it's very difficult to say, even based on that Osborne precipitation method. I'm not very sure you will get 100% albumins out precipitated. Do you get that one?
Yes, I get it, sir.
Okay,
so what
that's
would
good,
be
yeah.
the right method
Yeah.
to account how much albumins is exactly present?
Present in that, yeah, makes sense, makes sense. But as I said, the one important thing is, again, it depends on the sample. I might be, because there is albumins are also different types of albumins, maybe some precipitate, some does not. And as the good thing is that albumins are low. If you are working with seed storage proteins, albumins are low content, maybe you can precipitate them efficiently, and even if I lose some albumins, I do not care, because at the end of the day, food industries needs yield, and that yield usually comes from the globulin fractions. But of course, if you really want to understand the functionality of the albumin proportion, it is nice to follow this how to pronounce that one? Osborne's precipitation method carefully. Okay? Agree with you. And you can see I got this very nice figure from this very nice research review article. They have tried to combine the PIs of show the solubility profile of different well known plant proteins. One trend you can see, what is the trend here? Most of them globulins, and they can also see this nice curve, can you see? And the PIs or the least solubilities are always in this region, can you see? 4 to 6. So, most of the plant based protein isolates apparently shows PI 4 to 6, maybe because of globulins, which is also good, because you can use this isoelectric precipitation around 4 to 5, 4.5 or something, to get the most of the protein during the extraction. Am I clear? Now we will go a little bit about ionic strength, and we all know, I have taught you already this one. Yeah.
Sir, is there any method to extract albumins from the precipitate? Suppose we extract globulins by isoelectric point, then albumins may be present in the supernatant.
Yeah. One thing is you have to ask her, she can explain you what exactly she is doing, but what we do in our department is that we take the supernatant, we dialyze it to get rid of all the sodium hydroxide, salt, whatever if it is present, and then we do membrane filtration. So membrane filtration, you can actually, you know the albumin's apparent molecular weight, you do the membrane filtration and you get actually protein out. And that's a very efficient technique compared to precipitation method. So there are a lot of methods to get even protein from the supernatant.
Okay, sir.
Okay. Okay, now we go for the ionic strength. I have told you in my very first class, albumins are water soluble, globulins are salt soluble, prolamines are alcohol solubles. Okay. So remember this figure, salting in and salting out. I don't teach it again today, right? Is there anybody dare to explain me what is salting in and salting out? You remember the salt extraction? I told you protein solubility is highest at the low some concentration of salt, and then it decreases, right? And this region you will have more when you have protein. When you have another protein, the protein will have some charges here, right? When you have a salt, it shields the charges, avoids protein-protein interaction and increases protein-water interaction, and the protein solubilizes. When you have very high salt, all the water molecule will be taken by the salt to make a hydration, then the protein-protein interaction happens and protein precipitates. I have taught you already, I do not explain you this one again. So, you can use this phenomenon for the salt based extraction. Am I clear? So, here you can see that how different salt concentration affecting the solubility of bran protein concentrate. You can see that when you have solubility with water is this much. When you have a small amount of sodium chloride, solubility increases. When you have a very high amount of sodium chloride, solubility decreases, and this is because salting in, and this is because salting out phenomenon. I had explained that one already in my previous class. Okay. I do not think we should go this one. And the solvents also affect the solubility. If you add alcohol, protein precipitates, right, but prolamines will solubilize in alcohol, especially 80 or 70% ethanol you can use to extract proteins. Alcohol precipitates protein, you know, right, usually for protein precipitation in our laboratory, especially biochemistry people, molecular biology people, they add alcohol, because alcohol denatures the protein. Alcohol remove the accessibility of protein to water molecule, and protein precipitates because of protein-protein interaction. So, do not add alcohol if you want to increase the protein solubility, right. It will denature the protein, but if you want to extract prolamines, you need to use around 70% ethanol. Okay. I have some text that you can read, it is just an explanation, but whatever I just explained, it is already there. And coming back to your question, drying and freezing, and it has a huge impact on protein solubility, freezing and drying, because when you freeze dry for a long period, you remove the hydrated water molecules. It is very difficult to remove hydrated molecule, but they still say that water molecules which are on the surface of the protein will be removed, and then the protein is kind of a dry naked position now, and now that dry protein, that you have a protein powder, and then you did freeze drying or lyophilization, and then you get a very dry protein. And the dry protein basically having a structure that is so dry, it does not want water molecule now, right. There is a good intermolecular interaction now. It takes several hours to stir at room temperature or in the fridge to rehydrate the protein. So, if you freeze dry your plant based proteins, do not directly put into water and then just do vertex and then do not use it. It is a bad idea to do the research like that. Okay. If you at all you freeze or spray dried the protein, it is very important to rehydrate your protein minimum 2 hours, maximum several days. You need to do that experiment to see how much protein I can dissolve. 2 hours is usually minimum. I don't say it's universal fact, but I generally do minimum 2 hours stirring. But overnight stirring is recommended to improve the solubility. Then the protein will be hydrated and dissolve. Protein has a good solubility, but it does not like water any longer. If you stir it for a long period, it starts liking it and then it dissolves. Is it okay? Same thing is true with freezing. It's not recommended to freeze thaw proteins, because when you do the freeze thaw, small protein crystals will be formed inside the protein cavity between the particles, and that will denature the protein, ice crystals, micro ice crystals. It's okay to freeze your protein. You do lot of laboratory experiment, you can't finish experiment in the same day, right? You can't do extraction and functionality analysis the same day. So you need to freeze the samples, it's very normal. However, always I recommend that don't freeze it in a bulk big box. Freeze it in several tubes, right? You take the 1 tube per day for that particular analysis, right? Don't refreeze it again. Take the other tube for the next day, next experiment. Does that make sense? You freeze dry a big batch, and then you divide it into, you freeze a big batch, but you divide it into multiple tubes, and don't try to do freeze thaw. Freezing and thawing, temperature fluctuation will create ice crystals, and that can denature your protein, that can affect the solubility, that can also affect the functional property. But it's only to a minor level for most of the plant proteins, but it might happen. Okay? I hope I have a figure here. Yeah, I can skip that. Another one is present. Yeah,
I have a question.
Yeah.
So proteins generally denature after beyond 45 to 60 degrees Celsius, right?
Yeah, not all, in the sense depending on their denaturation temperature.
Okay,
yeah,
but generally.
Yeah.
So, but why don't we store it at the room condition?
You can always store it at room condition without any problem. In the dry form, the proteins are usually stable in a very long period. Most of the proteins, plant protein isolates are sold in a big bag, and they ask you to store it in room temperature, because the degradation of protein is very slow. But in solution, what happens, the reaction is different thing, because now there is a mobility of the proteins is very high in solution. They can move. When the protein moves, they collide each other. Now they want to react somehow, they want to interact somehow. That's the reason why it's not recommended to store in solution form in a long period.
I'm generally talking about the lyophilized protein. That we can store it in.
That's perfectly fine. I don't recommend lyophilized proteins. Again, you store it in the freezer. It depends upon what you want to do exactly. I'm a protein oxidation chemist. I know the oxidation happens at room temperature. The rate is high compared to minus 80 degrees Celsius. I want to know the thiol oxidation, for example. It's very sensitive to temperature. Then I would not store my protein in room temperature. I'll put it in minus 80. However, I want to do solubility study or functionality study. I don't care. I want my freezer space is more important than the 5% difference in the result. So what do you want to do? Based on that, you store your protein. Okay? Always lower the better, of course, I fully agree. But don't do freeze thaw. Okay? And the solubility is also depending on the other component present in the food. There is something called phytic acid. Have you heard? I'll talk a lot about that later in the other classes. It's an anti-nutrient. It contains inositol moiety with a lot of phosphate groups, right? And this phytic acid interacts with proteins and decreases the protein solubility. So one of the important point I want to tell you, almost most of the legumes or the seed proteins basically contains phytic acid. It is an anti-nutrient, it binds to metal ions. Phytic acid can also bind to protein, and they can affect the protein solubility. Okay, keep that in mind. It's often ignored point actually. We don't worry about phytic acid and solubility, but actually it can severely compromise protein solubility. So you need to remove phytic acid. How do we remove phytic acid? You can add phytase enzyme. Okay, so this is one of the experiments you can do in your also work. To improve the solubility, you can remove the phytic acid. All right, we will take a break after 5 minutes after this one. Then protein modification can also affect the protein solubility. As I said, proteins containing amino acids, and if I modify this amino acid with other things, the surface properties of the protein will be changed and the solubility will be changed. For example, if I modify the protein with the sugar contains OH group, maybe solubility will be increased. If you modify it with the let us say polyphenols, solubility can be increased, or sometimes solubility can also be decreased depending on what exactly happens to the protein. Lot of enzymes we do, for example, we can convert protein glutamic acid, glutamine into, we can convert into glutamic acid, and the glutamic acid gives a negative charge on the protein, and that can improve the solubility. So, we can try to change the, I will talk about this thing also in another exclusive lecture. Okay. And other thing is that if you have a protein, it is insoluble now. If you do a partial hydrolysis, it can become more and more soluble. You can use enzymes to do the partial hydrolysis, you can do fermentation to do the partial hydrolysis, you can add mild acid to do the partial hydrolysis. When you change the structure of the protein, the solubility can be changed, but you can also compromise the functional property if you do the hydrolysis, just to let you know. Okay. With that, we will take a break now, and we will come back in 15 minutes, 11:15. Okay. We will take a break, we will come back in 15 minutes. Hello, welcome back from the break, and again let us continue with the lecture number 7, functional properties of proteins, and we have been discussing about solubility. It was one of the most important functional properties to test in alternative proteins world. So, I took 1.5 hour-ish on that one. I was supposed to finish it in half an hour, but I will try to wind up the remaining lectures as soon as possible, so that I do not eat your lunch break. Okay. We still have to talk a little bit about water holding capacity, emulsification and foaming, but since I have given a very good background to you about solubility, the rest of the things will become much easier for you. I really wanted to spend more time on solubility because the other things are also a little bit related to solubility and the protein structure and so on. Okay. Let us start with water holding capacity, and if you look into any alternative based proteins paper, they do solubility analysis, then immediately they start doing water holding capacity. It is a common analytical methods we follow to characterize the functional properties of known or unknown proteins. So, the definition is not very clear for me, but you see if you do go into the internet and type, you will find different words: water holding capacity, water binding capacity, water retention capacity, imbibing capacity, adsorption capacity, and so many different things. At the end of the day, they are the same. They are interchangeably used. Okay. They might be, there is a little bit different meaning depending on the context of that particular paper. However, in generally I can say that they are all the same. Okay. So, nowadays most of the recent research article use the word WHC, water holding capacity. Okay. Is it clear? So, do not get confused with different terminology, they are relatively same. So, what do you mean by water holding capacity? There are a lot of definitions, but there is nothing about clear definition in food science. The water holding capacity is nothing but it is the ability of the protein to retain the water molecule which is bound on its surface, right. The ability of protein to hold water, that is water holding capacity, right. Ability of protein to hold water. And there is something like that, you have to keep it in mind that it is not simple holding. If you apply some pressure, if you apply some centrifugation forces, if you do some stretching and bending, still that water should be on the surface, and that is the determination of water holding capacity. Does that make sense? I have some nominal common definitions here. When you are free, just read through them, but in general it is nothing but the capacity of proteins to hold the adsorbed water on its surface or the hydrated water on its surface. People call it as it is something the water that cannot be freezed at minus 40 degrees Celsius. Okay. Now, if you remember about solubility, right, we had a beaker containing water and protein is added, right. Now, it is very simple to solubility also. okay, it is not nothing different than that one. So, when you have a protein here and if you have a water molecule have a fantastic structure, they have O and H, right. Remember that they have a delta negative and delta positive here, right. Protein is also a very complex molecule and it has lot of structure, like you have a carbonyl group sometime, oh sorry, it is a bad carbonyl group to write, it is on the peptide bond, but I will just show it in this way. We have a carbonyl group from the peptide bond, we have amino group, okay. We have carboxylic group, we have sulfur hydryl group, so many different groups we have. Some are charged, some are uncharged, and some we also have something containing ring, for example, phenylalanine, right. And they have some properties to love or hate water molecule. Do you agree? For example, we all know that the ionic compounds like water because of the ionic interaction, because they have a delta positive and delta negative, right. We also know that if you have a carbonyl groups, they are having the property to create hydrogen bonding, right. Oxygen is electronegative and then it can make a hydrogen bonding with water molecule. So, water tries to interact with proteins in different ways. Sometimes you have some ring structure, they do not like water molecules, they try to pull the water away. So, it is basically hydration of water. How does exactly it happens is that you have a protein molecule, the water tries to adsorb on the surface as a layer, okay, just to make it in the most simplistic way, okay. It adsorbs on the layer by layer. You have a first layer which is strongly adsorbed and then you have a second layer and you have a third layer and so on. Finally, you have a very loose layer. It depends upon the pulling capacity of the protein molecule. Protein is a chain, imagine, and you have water molecule is attached on the surface. It is basically the water holding. Can you see here? Please note that this water molecule, what I am pointing here, is different than the water present outside. This is the adsorbed water, the hydrated shell, and you have also water molecule here, and that is called bulk water. Is it clear? Two different types of water molecule I am talking about. Water that is adsorbed on the surface of the protein and water that is moving around the protein, they are different. The water it is adsorbed on the surface of the protein basically have a restricted mobility. It cannot move because it is attached to the protein now. But water is present around the protein inside the solution that can move. So, that is the difference between hydrated water and bulk water. Does that make sense? Usually the water holding capacity is determined by these particular types of water that is somehow attached on the surface of proteins. Am I clear? So, that is the water holding behavior. Why it is very important? Because we need meat which is juicy, right, and the juiciness of the meat is simply because of the water holding capacity of the protein in the meat. For example, in the true meat we have myofibrillar proteins and so many other proteins, and that proteins have a high water holding capacity, so that makes the meat juicy. Now, if you want to create plant based meat analog, that has to be juicy. In order to make them juicy, you need to add the proteins or create the proteins that is having little bit higher water holding capacity, because when you put them into the mouth that is released, you get that juiciness, right. And it should not be a bulk water, because the bulk water what happens is that when you remove the protein, the water molecule falls down. That water we do not want. We want the water, it is attached on the surface of the protein, and that is the water holding capacity, that is a hydrated water, and that gives you the nice texture, the juiciness to the food products you are going to create. Does that make sense? Clear. So, now I will show you a little bit about how the interaction happens. As I said, I mean I have already explained this one. Imagine this is my protein, the orange ones, right. The water molecules can be adsorbed on the surface like this one, and the bulk water molecules can be here, but we are more concerned about the water molecule attached on the surface. Okay. As I said, they can form layer by layer, that keep it in mind. It depends upon how many layers that can make, it depends upon the capacity of the protein to pull that water molecule, because the interaction could be because of hydrogen bonding, because of the ionic bonding and so many bonding. So, ionic bondings are really strong. For example, if you have protein, with lot of NH3 plus, right, then you get a more hydration shells like this one. Then if you have a protein with like you have let us say a peptide with the carbonyl group, basically they tend to make a one hydrogen bonding with one water molecule, something like this one, sorry it should be this way. So, the layer density can vary depending on the protein you have. That is the reason why the amino acid composition, the primary structure of the protein is very important. If you have more and more ionic amino acids, probably you will have a better water holding capacity, right. It also depends upon the folding of the protein, because if you have a more hydrophobic structure on the surface, it might have a less water holding capacity, it might have a more oil holding capacity, right, because the hydrophobic amino acids, they do not like water molecule, they just want to push. So, the folding is also important, but usually hydrophilics are surface exposed and hydrophobic are buried. So, they show you to a certain degree of water holding capacity. Does that make sense? Again, the water holding capacity is also dependent on too many parameters. For example, protein concentration, pH, ionic strength, temperature, presence of other components, and all these things. But these things I have already explained you in the solubility parameters. Exactly the same thing applies here. Solubility is nothing but the first step of water holding capacity. First water start interacting with this one and then it solubilizes. So, whatever the parameter that is applicable to solubility, usually the same parameters are also applicable to water holding capacity. So, I do not have to explain that in detail. Is it okay with you? So, for example, just to give an illustration, just to refresh your memory, how pH affects, you remember the curve. In the isoelectric point, there is a more protein-protein interaction, there may be different water holding capacity compared to non-isoelectric point conditions, right. Ionic strength, you remember the salting in and salting out effect, right. If you have some ions, they can shield the charge and they can change the water holding capacity, right. If you have a high temperature, that can denature the proteins, that will expose more hydrophobic residues, and then maybe water holding capacity can be reduced. Why? Because water does not like hydrophobic regions, it likes hydrophilic regions. Then the storage condition, it is basically during storage what happens is that protein can be denatured slowly, then you can change the water holding capacity. Then also important thing is that what is the other component attached to the protein, because they also determine the hydrogen bonding. For example, if you have a polyphenol attached on the protein and this OH group can also create a hydrogen bonding with water molecule, right. You can have a hydrogen bonding like this one and that can again change the water holding capacity. So, and that is the reason why in every single alternative protein research article they will measure water holding capacity, because they are very sure that depending on the processing, depending on the extraction condition, depending on the treatment they are doing, there will definitely some change in the water holding capacity, because the protein structure changes, water binding capacity changes, and the value what we call water holding capacity gram per gram, that will also change. Make sense? So, how are we measuring water holding capacity? How many of you measured water holding capacity? Perhaps you have done it, I do not know. How did you do, if you can recall? Yeah, do you remember? I mean, you do not have to, yeah, you?
I have taken 5 gram of protein.
Yeah.
I have dispersed, I have added water to it.
Yeah.
Then I have centrifuged.
Yeah.
I have taken the initial weight, that 5 gram.
Yeah.
After that, after centrifugation, I have decanted the water.
Yeah.
And again, I have taken the weight of the sample with water.
Yes, she took 5 grams of protein and she added how much amount of water?
5 ml.
So, she added 5 ml of protein, 1:1 volume here, and then she centrifuged or sorry shook, vortexed or something did you do? So, she vortexed. Did you wait for some time?
Yeah.
Yeah, you must have waited some X minute, it is okay. And then you basically get a suspension here, right? You get a suspension and then you centrifuge that suspension and you get a pellet and the supernatant, and you carefully discarded the supernatant, and then what you get the pellet. Okay. I think you have weighed this one, did you say that one? Yeah, so you took the weight of this one before water, before adding the water, right? You took the weight of this one before adding the water, and you took the weight of this one after removing the water, and the difference divided by the original weight will give you the water holding capacity in percentage or gram per gram. Does that make sense? This is the most common method that we use today. I also copy pasted this method from one of the research articles to show you, you can use this method without any problem. The idea is simple. You have a protein, you add an X amount of water, right? Then the water will go and bind to the surface of protein, as I mentioned previously, in a different layer, right? And then you centrifuge. When you centrifuge, what happens? The hydrated water, the bind water, the adsorbed water will not be removed. Am I clear? Because they are tightly bound on the protein surface, but those free water, the mobile water, will go away because of the centrifugal force. When the protein precipitates, it will bring protein along with the hydrated water, but it will release the non-bound or free water. Now, if you take the difference in the weight, that will give you the water holding capacity. Make sense? It is a very simple experiment to do, in my opinion. Often people define in recent literature, water holding capacity is nothing but amount of water bound to per gram of protein. It is a normal definition in the recent article. How much amount of water in gram can be bound or adsorbed or hydrated on per gram of protein is called water holding capacity. And that is a table you will see in every research article. I can show you one example here, just to compare, just ignore this one for now. Just to compare different, this also you ignore, I will come back to later. Soy protein isolate, pea protein isolate, fava protein isolate, I think it is lupin or no, lentin protein isolate. So, you can see the difference in the water holding capacity. I have been keep praising soy protein, right. Soy is extensively investigated simply because soy has every single functional properties in a very good level compared to any other plant proteins. You can also see that it really has a very good water holding capacity, and that is the reason why most of the product sold in the US market at the moment containing soy, especially the plant based analogs, meat analog, chicken analog, beef analog, egg analogs, everything contains soy. It is simply because that gives you this juiciness, because it contains, it has a really good water holding capacity equivalent to the common meat based proteins actually. So, that is really cool. Am I clear? The factors affecting the oil holding capacity, you need to be while measuring this oil holding capacity, you need to keep in mind of these things that often people ignore. The first one is protein concentration, because if you change the protein concentration, oil holding capacity, water holding capacity will change, right. And centrifugation time and the G value also important, and hydration before measurement is also important, because she mentioned that she vortexed and she kept it for some time. If we kept it for a longer time, you will give more time for hydration, and then the water holding capacity will also change. So, there is again, I have been reading some article, is there any uniform method people are following? Am I correct? No methods. Everybody follows their own protocols depending on the type of centrifuge they have in their laboratory, to the best of my knowledge. If their centrifuge cannot run for a faster speed, they can't do that one. So, that is a problem. So, there is no uniform method here. Again, we need to have a standardized method to measure water holding capacity, and that is the reason why I really enjoyed reading this particular paper. I think this is from Julian McClements from University of California, Davis, it's from Davis, I think it's from Davis. He made a really nice comparison comparing 4 different commonly used proteins and how they show different functionalities using the common analytical method. It's very difficult to compare between paper to paper. We can compare the data within the paper. So, they compared it and they can very clearly shown that soy protein isolate has a really good water holding capacity. This is something the paper the table I tried to make. I am not very sure this data is absolutely correct. I do not trust this data. I am telling I do not have a citation because I am trying to read many articles and try to compare. But we can say that the plant proteins and the milk proteins have somewhat similar water holding capacity, which is really good. That means we can use plant proteins as an alternative to dairy proteins in terms of water holding capacity. Okay. I am repeating, do not believe this absolute number. I am not very sure because it is just a range I am trying to write. I do not have a citation for this one. Is it clear? So, the take home message here is that especially soy protein is an excellent replacer of meat and dairy proteins, but soy has its own problem. Remember, sustainability problem. So, we need to somehow change the properties of P so that it can replace soy, okay, or use insect or use microalgae. Please note that I have been talking about plant, I am not including insects and microalgae in my slides. It is simply because we do not have a lot of data on them. We do not have data only on extraction and basic functional properties, not on in-depth functional properties comparing with plants or animals. Okay. All right, now we will little bit move on to the next important topic, oil binding capacity or oil binding properties, which is exactly similar to water binding properties, but an opposite one. Do you agree? Oil and water. Oil has a hydrophobic carbon side chain, triglycerides, right? They are opposite because oil is non-polar. Do you agree? Water is polar. So, in the case of protein, what happens if you have a polar residues, let us say amino group, it likes water molecule, right, because polar interaction or ionic interaction. But if you have a non-polar residues, let us say leucine or let us say benzene rings and so on, they do not like water, they like oil. So, basically instead of getting the water layer, you will start getting the oil layer on the surface, and the capacity of the protein to hold oil is the oil holding capacity, just opposite to the water holding capacity. Is it clear? So, the protein can have both oil holding capacity and water holding capacity, because proteins are kind of amphoteric. That means it has both not amphoteric is a bad word to use, sorry. It has both polar region and it also has non-polar region. It has hydrophobic region, it has also have hydrophilic region, and that is the reason why the protein is an excellent oil binder and also a water binder, right. It has a dual properties. It varies depending on the composition of the amino acid plus folding. Usually denatured proteins can have a better oil holding capacity. Why? Because you expose the hydrophobic region and the oil can bind nicely and you get a higher oil binding capacity. I really want to tell you one thing is that the processing impact the oil and water holding capacity. That is the reason, because you denature or renature the protein depending on the processing, heat treatment, and the purity also matters. If you have an impure proteins, it may have a different oil binding capacity compared to pure proteins. Why? Because the other components present in the proteins can also interact with oil, okay, or water. So, the oil binding capacity can change. So, be careful about that when you measure the protein purity, and the condition and the concentration, pH and everything matters while determining the oil holding or water holding capacity. How do we determine water holding, oil holding capacity? Same as previous experiment. Instead of adding water, add oil. What type of oil do you use here? Do you remember any type of oil? Which one did you use? Organic sunflower oil. That's nice to use. Okay. In fact, I have seen papers showing different oils. Some people use So, rapeseed oil, some people use organic sunflower oil, some people use sunflower oil, some people use soy oil, different oils have been used to measure oil holding capacity, and there is no universal common oil to be used. It depends upon which oil you want to actually finally you want to interact it. So, you are welcome to use the oil you want, but if you want to compare it with different papers, try to use the same method and same oil, then you can make a comparison data. Make sense? So, again there is a lot of factors affect the oil holding capacity in it similar to the water holding capacity. I do not explain that in again, but one of the things you have to think about while measuring the oil holding capacity, what you do is that you take a beaker, you weigh the protein 5 grams what she meant, and she added organic sunflower oil, okay, and she measured then the oil holding capacity. It is very important to know that especially if you use proteins coming from the seeds, leguminous seeds, they already contain some oil. Keep that in mind, because depending on the extraction, whether they have used air classification or whether they have used hexane extraction, depending on that, there is always a trace amount of oil already present in the protein, and that might actually affect the oil holding capacity. So, the true oil holding capacity can be compromised if it is already having other oil. Make sense? So, it is very important to keep that in mind when you measure oil holding capacity, but it is very difficult to remove that oil present. There is a big challenge, because to remove that oil you need to do hexane extraction. If you do hexane extraction, you will denature protein. If you denature protein, oil holding capacity will change. We are in a problem now. So, it is very difficult to do, but still keep that in mind. You need to measure how much oil is already present in the protein before measuring the oil holding capacity. Is it boring? I know it is boring, but it is an important experiment to do. I am just trying to explain. Okay. Now, I will show you a little bit about curves which I wanted to mention, how different factors affecting the oil holding capacity. I found this from this book, and they show clearly that increasing the protein content, oil holding capacity increases, but the temperature has no effect, okay, especially for oh my god, I forgot to mention which oil it is, which protein it is. I think it was from pea or sunflower. I think it is from sunflower protein isolate, but we can check it in this paper. In case of wheat germ flour, again they see that increasing protein concentration increase the oil holding capacity, but the temperature has a very minimum effect. But it is not always the case. For example, in this case of corn germ oil, increasing protein concentration increase the oil holding capacity, and at a particular temperature they found highest oil holding capacity. So, temperature does matter, and that varies depending on the type of the protein, okay. But in general, if you increase the protein concentration, you will definitely get more oil holding capacity, okay. Now, this is a figure that is showing how different processing affect oil holding capacity. You can see that this is sonicated samples, this is non-sonicated sample. Sonication is ultrasound treatment. You can see that when you do ultrasound treatment, oil holding capacity increased. Can you see? When you do the when you do the ultrasound treatment, oil holding capacity got increased. It is simply because probably ultrasound denatured the protein, probably exposed to hydrophobic moieties, maybe oil is more interacting with the protein surface, so increase the oil holding capacity. What I wanted to tell you here is that the processing matters. Depending on the type of processing you are doing, the oil holding capacity and water holding capacity can change, okay. Here is another example of how different proteins and their oil holding capacity from the same paper. You can see that soy has the highest water holding capacity, but it also had a very good oil binding capacity. That is the beauty of soy. It is an all-in-one protein in plant-based world. It can do many things together, many jobs simultaneously. But in this case, if you ignore the standard deviation here, no, there is a statistically insignificant, you can see that everything. So, they are all having the similar oil holding capacity. Make sense? Now, we will move on to the emulsifying properties of proteins. How many of you have done emulsifying properties of proteins? You have done it, good. Have you heard about emulsion? Can you give me one example of emulsion in your real life? Huh? Mayonnaise, right? Do we use mayonnaise a lot in India nowadays? Yes, especially in the burger and they add, right? Exactly. But it's not in our traditional cooking, right? Do we have mayonnaise? Milk, yes. Butter, yes, exactly. Butter is a good example. And while we make omelette, you take egg and you shake it and then you add tomato and then you create a nice emulsion there, right? Water and oil emulsion. Remember omelette making? Remember? Right? It's a good emulsion according to me. So, any other example of real life oil emulsions? There's a lot of examples. Now you know you are becoming more food scientist. Whenever you go back home, think about food carefully. Think about which one is emulsion, okay? Now, a simple thing, oil and water does not mix. Agree? Oil and water never mix. In normal temperature and pressure condition, they never mix. But you take oil and water and you shake vigorously, right? For a couple of seconds at least you see they are kind of mixed, not really mixed, they are kind of mixed. Do you agree? You start creating kind of an emulsion because of this vigorous shaking. What exactly is happening here? Oil droplets can be broken down into small pieces, become a micro droplet because of this vigorous shaking, and it will try to disperse within the water phase. Do you agree? Because in this case at least if you imagine you have a higher volume of water and lower volume of let us say oil, and when you shake it like this, you get a small oil droplets within the water phase and it creates kind of an emulsion. In this case, if you have more oil and less water, basically within the oil phase you will get a small droplets of water. So, water in oil emulsion. So, by shaking you can start creating the emulsions. Am I clear? In real life. Now, by definition, emulsion is nothing but dispersion of two or more immiscible liquid, that is very important, two or more immiscible liquid, one of which is dispersed in the other as a small droplets. Okay? It's basically you have two immiscible liquids, this is liquid number 1, L1, and the L2 is a small droplets. This is L2, okay, and that is called emulsion. Am I clear? Both are immiscible, but the other one is dispersed in the another one, basically dispersion of one in another as a small droplet, and that is called emulsion. You can, it's a nice example I got. These two things are not emulsified, but this one is really emulsified. Can you see? And when you add an emulsifying agent, you will create a nice emulsion. It's a stable one. It is very, emulsification is extremely important. Why? Because we would like to use this plant based proteins or insect proteins in solution in many cases. We want to create sports drink. We want to create liquid infant formula. We want to create yogurt. We want to create milk. And after creating them, we would like to put it in the supermarket shelf. If you have a phase separation, people won't buy that one, right? They really want something in a single phase. If you have a mayonnaise, for example, mayonnaise bottle you buy from the supermarket, if you have an oil phase and if you have a water phase on a mayonnaise, do you buy it? You think it something is wrong with this and I don't want to buy. It has to be really a good single phase, a good emulsion, and that's the reason why emulsion and emulsifiers are very important. Emulsion is important to get a structure, a good taste, good texture, sorry, and emulsifier is important to stabilize the emulsion. Am I clear? Now, I will talk about two different types of emulsions, oil in water emulsion and water in oil emulsion. It is very simple. As the word says, in case of oil in water, this is oil in water, water is the bulk one, oil is the droplet. Am I clear? In water in oil, water is the droplet, oil is the bulk one. That is it. Oil in water emulsion and water in oil emulsion. There is a lot of examples. For example, milk and mayonnaise are all oil in water emulsion. Margarine and butter, butter has a lot of fat, so it is basically water in oil emulsion. These are the normal examples. Any doubt about these two things? Okay, we can have two types of emulsions, oil in water and water in oil. In case of oil in water, the droplets are, the micro droplets are oil, and water is the immiscible liquid here. Now, how do we create emulsions? As I said, the easiest way is to take two immiscible liquid and shake it, you get the emulsion, but they are not stable. Within a minute, within a half a second, they get a phase separation, alright. To avoid this one, we do homogenization, because the homogenization will help us to create micro bubbles or micro droplets, and it will create the homogeneous micro droplets in the bulk of oil or water. So, homogenization is one of the common technique used to create emulsion. Do you remember ultra 2x in the laboratory? You can also use ultrasound, but ultra 2x is one of the efficient way to create emulsion. I mean, as I said, you can take a spoon and do it like this one, but the problem is it is not efficient. We need to put a very high shear rate to the emulsifying liquid here to create these micro droplets, and to get that one we use an equipment called ultra 2x, okay, and that can create these micro droplets like this one within the within water or within oil, depending on oil in water or water in oil, okay. We also use one of the equipment called high pressure homogenizer. Have you used it? It is also one of the most efficient instrument to create emulsions, okay. High pressure, this is the one equipment, we also have it in our own lab. What we do here is that we take these two immiscible liquid and we put it into this particular chamber here, and inside this one they actually pass through a very narrow space at a very high pressure. We are talking about pressure, if I am not wrong, maybe more than 200 or around 2000 bars, it's super high pressure, and because of that high pressure and homogenization, a micro bubbles are formed. Just to show one example here, we put the product in here, right, and they are mixed here under a very high pressure, and they are passing through the very narrow chamber, right. This is illustrated in more here. You have oil droplet, for example, now it is a bigger in size, can you see, right, and they are passed through the narrow chamber, and then they have a Y splitting, they split it into two different tubings here, and they come back in this region in a very narrow space, and they collide each other. When they collide each other, they create a very shear stress here, right, a very shear stress. Because of the collision of the droplets, you get a micro droplets, right. Now, the bigger droplet becomes smaller droplets, then you get a very fine emulsion, and this is one of the most commonly used method to create a very stable or a homogeneous emulsions, okay. We use high pressure homogenization, HPH, okay. Now, we talk little bit about the emulsifiers now. Now, if I talk about emulsion, let us say oil in water emulsion, this is oil droplets and this is water and this is oil, okay. Now, we need a stable emulsion, do you agree? The emulsion should be stable. That means, if you put it in the supermarket shelf, if you put it in your kitchen, we do not want to have a phase separation. We want them it homogeneous as much as possible. But it is very difficult to have a homogeneous emulsion in reality. We need to stabilize this emulsion, because the droplets, they have a crazy behavior. When they have a small droplets, initially because of the homogenization, you create a small droplets, but these droplets can interact with each other, because they are very so close to each other, they interact with each other and they can create another new phase now. we do not want to have the surface interaction between the droplets, to avoid that one. So, to avoid this all interactions, to create this very stable emulsion, we add a emulsifier. Okay. Emulsifier is nothing but that is a reagent, it is a chemical that stabilizes the emulsion. It will also help to create smaller droplets. I will show you how. Emulsifier has two properties. It has both hydrophilic and hydrophobic tail. It is amphoteric in nature. It has both hydrophilic moiety and hydrophobic moiety. Okay. In case of, let us say, oil in water emulsion, you have an oil droplet, right. Oil is non-polar in nature, right, and this emulsifier will place in such a way that its hydrophobic end will attach to the oil and hydrophilic will go to the water phase, right. Hydrophobic attached to the oil and hydrophilic go to the water phase. So, it nicely surrounds the oil droplets nicely. Now, since it surrounds the oil droplet, if I have a new oil droplet, now imagine here, it is a new oil droplet, but the surface of this oil droplet is hydrophobic. Do you agree? It is hydrophobic. Now, the surface here is hydrophilic, can you see? And they do not interact each other. While you are creating this, when you are doing homogenization, if you add an emulsifier, oh sorry. So, when you are doing homogenization, when you add emulsifier, basically when the new droplets are forming, it will avoid the interaction between the droplets because it will create a barrier, can you see, right. Not only that, it covers the surface of the droplet, it will stabilize the droplet. So, the droplets will not interact with other droplets, because now this is hydrophobic and this is hydrophilic and this is hydrophobic, there is no interaction. Am I clear? And that is how it will start helping you to create very small droplets in the homogenizer. The opposite is true in case of oil and water emulsion, where the water molecules are hydrophilic. So, the hydrophilic region of the emulsifier interacts with the water droplet and the hydrophobic region interacts with the oil, and that is how the emulsifier will stabilize the emulsion. Are you getting what I am telling? Are you sleeping? I know it is getting late now. You are getting my point, right? What is emulsifier? Emulsifier is a reagent, it will help us to stabilize the emulsion. It will also help us to create small micro droplets. Okay. Now, I will tell you how protein acts as an emulsifier. Okay. How protein has an emulsification behavior. Let us explain how protein acts as an emulsifier. Now, let us take this in water in oil emulsion. There is an example here. Now, we have a bulk of oil and we have a bulk of water, and this is oil droplet. Okay. Now, I will add a small amount of protein, plant protein for example. Now, I added a small amount of protein. Protein is 3-dimensionally folded. Okay. Protein also has a behavior like a true emulsifier. It has a hydrophilic, it also have hydrophilic region, right, because protein is made up of amino acids. Some are hydrophobic and some are hydrophilic, right. Some contains OH group for example, and that gives the protein also dual properties, hydrophilic and hydrophobic. Now, you add that protein to water containing oil droplets, right. Now, what this protein will do, if it has a very good emulsification properties, it will slowly migrate towards oil, the first step. Okay. It will migrate towards the oil, and then it will start orienting in a way that the hydrophobic region of protein will interact with the oil and the hydrophilic region of the protein will interact with the water. That is the second step. First step, diffusion, migration. Second step is orientation, where it will orient in a way that hydrophilic region of the protein will interact with water, hydrophobic region of protein will interact with the oil. Okay. Then it will slowly denature, okay, and oh, I'm sorry, maybe the denaturation is the first and then the orientation. It will denature, then it will orient nicely. And the last step, it will cover the oil droplets nicely. Okay, it will cover the oil droplet nicely, 3-dimensional, exactly in this way. Okay, and that's how it will stabilize the proteins. Is it clear? How protein acts as an emulsifier? Okay, in oil and water emulsifier, protein will go slowly. It will attach on the surface of the oil droplet. It will denature it, and it will orient properly in a way that hydrophilic region go to water side, hydrophobic region go to oil side, and that will create a stable emulsion, because now protein will surround the oil droplets as a thin film, and that will help in emulsification or stabilization. Do you have a question? Yeah. The water bottle. Yeah, yeah. Yeah, go on. I think I dropped my water bottle there. Can you get my there? Yeah.
Sir, is denaturation important for protein to act as an emulsifier?
It's a very good question. Denaturation is important and may not be important. Depends upon, usually what happens, the denaturation is helping in exposing the hydrophilic and hydrophobic region. What happens exactly is that when the protein comes and attached to the oil body, it tries to attach to the hydrophobic region of the protein, all right, and then the water molecule start pulling the hydrophilic region, and the protein will slowly denature, and the denaturation will actually help the stabilization of the protein. Yes, good question. The driving force, I've been reading the same question, what is the driving force here? Apparently it says that it's simply an interaction, because once protein attached to the oil surfaces, now it will try to make a new hydrogen bonding with water surface, right. New hydrogen bondings are created, and that will pull the protein apart, and that will leads to the denaturation. It's all surface property, interfacial properties. The driving force is basically formation of new hydrogen bonding and hydrophobic bonding, because now the protein is oriented like this one. Okay, now you have a oil droplet. Now it will come and attach to the hydrophobic region, right. Now the original hydrophobic region is gone, because it now attached to the oil region. Now the protein is oriented in such a way that now we have a water molecule outside, it exposes the region, so it will make a new hydrogen bonding in a particular orientation. Apparently that releases that makes the denaturation of the protein. It's all very slow and what we say, a minor processes. I don't say it will completely denature to a minor level.
Will it differ between the two phase, like oil in water and water in oil? Will it differ?
Yeah, it will differ. In oil in water emulsion, the hydrophobic region will orient like this one. In water in oil water is a reverse of that one.
They are having hydrophilic nature outside
yeah.
But if we add them in oil in water,
yeah.
so then there is
absolutely
there is a hydrophilic nature in both sides.
Yeah, it is yes and no, because one of the properties of protein, that if you want to use it as an emulsifier, it should not be completely hydrophobic. Then it will start dissolving in the oil droplets, and we don't want dissolution. We just want a surface interaction. Okay, that's a very important property of a protein, that if it is completely polar, it doesn't act as a emulsifier, it just dissolves in the water phase, right. If it's completely non-polar, it just dissolves in the oil phase. We want a protein which is slightly polar and slightly non-polar, and that is the only way it interacts. Am I clear? So that's one of the that's a protein property, you have to keep it in mind. It should not dissolve in any of the phases, it should just orient. Okay, yeah. Now factor affecting the emulsification properties. So we have a pH that has an effect, and protein concentration has an effect, exactly the same thing what I have explained in previous slides. In isoelectric point, they have a more aggregation, and if you have an ionic, if you have positively charged proteins in a different pH, they migrate in a different way. If you have a negatively charged protein, they migrate in a different way, they orient in a different way, they create a hydrogen bonding in a different way. So, pH affects the emulsification properties. It is often people measure that when how pH shows the emulsification index. Same thing is true with the ionic strength, which I have already explained in the solubility. Now, I have one example here, the emulsification capacity of whey protein, which is a milk protein. So, you can see that depending on the protein concentration, it increased and then it decreased, right. You need to find out the right protein concentration to create the emulsion, otherwise it will destabilize the emulsion. Please note that when you talk about the emulsification, we need to add a small amount of protein. If you have overloaded the protein, what happens is that it creates a new phase, it is no longer an emulsion. You start precipitating the protein, so you destabilize the emulsion. So, the critical emulsification protein concentration has to be determined. Okay. And please note that when you want to use protein as an emulsifier agent, it is not for the nutritional purpose, it is just for the emulsion purpose. It is not about adding more and more proteins, it is adding the right amount of protein get the emulsification properties, not to get the nutritional benefit. Okay. And here is the emulsification properties of different soy types. They have a soy flour, soy isolate and soy concentrate, and this is the isolate, this is the flour, this is the concentrate. What did these guys found was that temperature had a minimum role on this one, and also the very interesting point that they say that even though they have a different purity degree, they did not see a much difference in the emulsification property, but you can always compare the y axis here. Flour had a little bit higher than the concentrate, and the isolate also had a very high emulsification property. So, depending on the degree of purity, you get a different emulsification properties. Okay. But I do not want to cover these 2 points, but you have read it in a research paper. They talk about emulsifying activity, emulsifying capacity, emulsifying stability. Emulsifying stability is nothing but how stable is emulsion during storage or different treatment. Okay. Now, how do we measure the emulsification properties? There is a lot of techniques to measure emulsification properties, but we often use in alternative world droplet measurement, right. Finding out the droplet size, because whenever we say emulsification, it is basically formation of droplets, right. The droplets can be of different size. The number of droplets and the distribution of the size of the droplet talks about the emulsification capacity or efficiency here. Okay. Total number of droplets plus the size distribution of these droplets within the emulsion talks about the emulsifying properties, and the size distribution of the droplets can be measured using what we call master sizer. Basically is it measures the particle size based on the light, because the light will be, I do not know if it is a refraction or diffraction phenomenon, I forgot completely now. The light will be, the property of the light will be changed based on the, is refraction? Diffraction? Diffraction. So, the property of the light will be changed based on the size of the particle, it diffract in a different ways, and you get a different particle size distribution, and that can be used to measure the emulsification properties. Okay. Here is one nice example I found. They show that how different protein concentration affects the particle size distribution. You can see that increasing the protein concentration decrease the particle size, right. Increasing the protein concentration decrease the particle size, so the droplet size. And they are also trying to compare different protein isolates and their droplet size. You can clearly see that which one had the highest one. In general, the gray one showed the highest biggest particle size, it is fava, right. You can see that the soy, which is the light gray, this one, it gives the small particle size. And the beauty is that when they use the lentil protein isolate, so that is over here, that gives you the lowest particle size. That's really good. We can say that the lentil protein isolates can be used to get lowest particle size. But please note that it's very important to know the protein concentration as well. We don't want to add a lot of protein into to get the emulsification property. No, that's not the idea. We need to use a small amount of protein because then it's sustainable. I'm telling you again, we are not adding protein to get a nutritional benefit. No, we are adding the protein to get the functional property here. So it's better to add a small amount, then you save money, right? In that sense, I think this paper shows that the lentil is the best protein to use. If you really want to study emulsification properties, you can go for reading this particular paper. They have a lot of information about the emulsification property. We are going to take a break now. So, we will take a break now, and we will go back to the next lecture. Okay? Is it okay? I will definitely send you by 12:45 to lunch, do not worry. So, we will take a break now. In the break, we are going to talk about a little bit about the foaming properties, and then I will talk about the last lecture of today. Okay? We will take only 10 minutes break at the interest of time. Okay? Only 10 minutes break, and we will get back. So. So, welcome back, and we are still on lecture number 7, functional properties of proteins, solubility, water holding capacity, emulsification and foaming. We have covered the solubility, water holding capacity and emulsification as slowly as possible, and we are left with the last one, foaming capacity, and forget about the timings here. We are actually delayed by more than 1.5 hours according to me, but it, as I mentioned, it is an important topic. I am trying to cover the fundamentals here. And the class will be like this. I will take about 15 minutes now, and I will talk about foaming properties, and around 12:45 I will leave you guys for the lunch, and then we will resume back at 2 o'clock, and we talk about the remaining 2 lectures. The one is about how are we using plant based proteins to get meat analogs, and what are the different technologies we have, for example extrusion and etc. That is the last lecture, and those 2 remaining lectures I will cover in the afternoon session. It will take about 1 hour. Okay? So, about 2 to 3, let us say like that. So, now I will only take 15 minutes for foaming. Foaming is important. You know chai? We just had chai, right? And when when you look into this YouTube reels, the chai walas, they make like this. Have you seen? Right? Why do they do that one? They get the foam, right? The foaming is an important functional property. It is an aesthetic property. We want to see that foam. And Horlicks advertisement, have you seen? You drink and they get something on this here, right? The foam, right? If you come to Europe and Americas, cappuccino is one of the most sold beverages, and that has a foam. They bubble hot air, I do not know, this like hot milk to coffee, and then you get this foam. So, the foaming is very important. Ice cream also has some kind of foam, right? It is an important functional property. I do not know how, for me it is not important, to be frank. I do not care whether chai has a foam or not, I just need chai, but for some people foaming is very important. It is an important functional properties. What do you mean by foam? Basically, foam is nothing but it has air bubbles. In emulsion, we have oil bubble or water bubble, right? So, here we have an air bubble, right, and there not a bubble in that, it is a droplet. Here it is actually we have an air bubble, and that air bubble is actually covered by a thin layer of liquid. It could be water in this case, right, and that is foam, right. Foaming capacity is something very important, because once you make this chai like this, right, and he get it in the glass and he will bring and give it to you, right. You should have that foam at least until you start drinking, right. If the foam disappears immediately, then there is no use of foam, right. That is what we call foaming stability. Two things are important, foaming capacity and foaming stability. Foaming capacity is how much foam can be formed, right. We should not have too much foaming also, because then basically you take Coca-Cola for example, right. You go to McD or you try to take a Coca-Cola, if you pour it so fast, you will only get little bit Coke and too much foam. We do not want that also, right. And we need to find a fine balance between foaming, and the stability is important, means it should be stable for some period so that the consumer can consume it. If it disappear immediately, there is no use of foaming. That is the reason why capacity and the stability of foam is important. We are going to talk a little bit about foaming, how exactly foaming forms. It's very similar principle as I just explained in the emulsification. What exactly happens here is that when you take, let us say, water and about small amount of protein, let us say in this case 10%. So, you have water, this is water and these are the proteins, right. And then you do whipping. Whipping is like shaking or doing like this one or bubbling air, whatever it is, right. When you supply high flush of air, whipping, then basically there is a formation of air bubbles. Can you see here, right. Now, we have three things here. One, we have water, we have protein and we have air bubbles, right. Now, protein has again this property. It has hydrophilic region and hydrophobic region, right. Hydrophilic region likes water, hydrophobic region likes, in this case, non-polar thing, it is air, right. It slowly diffuses towards the air bubbles and it will orient in a way that the hydrophobic region will go towards air interface and hydrophilic region will go towards the water interface. It will slowly cover the air bubble, right, and thus it will stabilize the foam by stabilizing the air bubble. But does that make sense? It creates the thin layer around the air bubble upon denaturation of the protein. Very similar to the mechanism I explained to the emulsification. In the emulsification, the surrounding of the protein happens on the bubbles, the droplets, right. In this case, it surrounds the air bubble, that is the only difference. Okay. How do we measure foaming properties? There are different methods, but at least in a food science, we use this common approach. We take the measuring cylinder and we add water and we add our protein and we can supply air if you want, or you can shake what you want, but usually we supply air, right. We supply nitrogen or carbon dioxide and then the foaming occurs. Then because of the foaming, the volume of the foam increases in the measuring cylinder, because it foams, then the foam increases like this one, right. Then if you have a time versus length here or percentage of or volume, okay. Initially you get a high volume of foam until here, right, maybe 80 ml, let us say. Over the time, slowly the bubble collapses, right. Then the foaming decreases like this one. After half an hour, after 10 hour, after 30 hour, after 3 months, I have no idea, depending on the stability of the foam, this curve goes down. Am I clear? If we have a very stable foam, this curve, the time will take forever, okay. If you have an unstable foam, it will drop quickly. Does that make sense? That is the foaming capacity. This is foaming capacity, this is foaming stability. How long it can go in terms of volume is foaming capacity, how stable it is in terms of time is foaming stability, and that can be measured easily with this flask experiment. Again, it depends upon the air bubble the air you supply, the length of the breadth of the measuring cylinder, a lot of parameters, and I did not find any research article they follow the same method. Everybody have their own methods here also, but you are also welcome to follow your own method, but report carefully what exactly you have done. Am I clear? So, I have some examples about foaming stability and how that is varying with respect to pH. You can clearly see that they have high foaming stability at pH 6 compared to 7 compared to 8. I would like to mention that pH matters here also in the foaming, it's simply because pH changes the three-dimensional structure of the protein. When protein denatures or renatures, its surface properties changes, how it covers around the air bubble also changes. Depending on that, you get a different foaming capacity and foaming stability. Okay. And here is a nice example of different plant proteins and their foaming capacities. Let us look into the y axis here. You can see that the cowpea can go up to 90-ish, and peanut can also go up to 90-ish, soy can also go up to 90, 95-ish. So, all the plant proteins or most of the plant proteins really have a good foaming capacity. It is more about foaming stability, how fast they drop, and that varies depending on type of processing, right. For example, if you have pure proteins, they might have a different foaming capacity. If you have a partially hydrolyzed proteins, they might have a different foaming capacity and different foaming stability. We often do proteins and we hydrolyze into small fractions, and they might have a different foaming capacity. So, basically depending on the processing condition, you can change the stability as well as the capacity, and there is nothing called that you need to have the best foaming stability. Best foaming stability is also sometimes problematic. We need to have a balanced foaming stability to get the desired foaming properties for the given food right. All right, and that is it for the 15 minutes I have been talking about foaming. So, what did we do in lecture number 7? We started understanding about different properties of proteins, especially the functional properties. I began explaining you about solubility. Before that, I mentioned to you that structure is important. Everything is determined by the structure of the protein, both primary structure and the secondary and tertiary structure. We talked about protein water interaction, mainly solubility and water holding capacity. In case of solubility, we understood that how protein solubilizes in water depending on the structure of the protein, amino acid composition, and so on. We understood different parameters affecting the solubility. We talked about how pH affects the solubility, how salt affects the solubility. We also talked about how globulins are different than albumins. Globulins have a very beautiful curve for pH, albumins does not have, and that is an important property, we have to keep it in mind. Then we slowly move towards how water holding capacity varies with respect to different types of proteins. Water holding capacity is very similar to solubility properties, but here in this case how strongly the water can adsorb on the surface of protein, that we can determine. We discussed about different methods, and we realized that everybody uses different methods, but at the end of the day we try to measure the amount of water in grams holded by gram of protein, per gram of protein, gram per gram, that is what we measure. Exactly same thing is for oil holding capacity. Instead of water we use oil in here, right. Then we went to which property? Emulsification property, and we talked about protein can have both, stabilize both oil in water and water in oil emulsion depending on the polarity and the structure of the protein. Protein can act as a fantastic emulsifier, and soy protein isolation is one of the best emulsifier today in plant based world, right. Then we came to foaming. Foaming is very important to get the foam for the desired beauty on the food product, especially in beverages, and it is nothing but air bubble is now covered by the protein to stabilize the foam. Both foaming capacity and foaming stability is important to use it in different food products, especially in the beverages. That is all about chapter number 7, and in the afternoon we will move to the chapter number 8, application of alternative proteins in plant based meats and dairy alternatives, and chapter and lecture number 9, which is about extrusion technologies. All right, so now we take a break and we will go for the lunch break, and we will come back at 2 o'clock in the afternoon. Enjoy your lunch. Okay, I will take about 1 hour in the afternoon. Thank you so much. See you later. Hello, welcome back. So, in the last session, we have been discussing about different functional properties of proteins like solubility, water holding capacity, oil holding capacity, emulsification, and finally foaming, right. I was telling you that usually plant proteins or insect proteins may have one or more these properties. It is not necessary that a single protein can have all these properties. And these properties are important to be measured. It is simply because we can use these proteins in one or other applications. We can create new foods, we can create new food structures, we can create new food flavors, we can create new texture based on all these properties. So, that is the reason why we need to measure functional properties of proteins when they are present in individual form, right. Please note that in the last session I did not talk about the whole food, right. I was talking everything on an ingredient basis, which individual ingredient, protein ingredient. For example, we were talking about soy protein isolate, soy protein concentrate, pea protein isolate, they are all ingredients. I was also mentioning to you in my previous lessons that ingredients cannot necessarily be used directly because of their less taste or no taste. So, we need to again functionalize them with other techniques and technology. So, that is we are going to hear now. So, welcome back to the lecture number 8, that is application of alternative proteins in plant based meats and dairy alternatives. And I must tell you that some of them I have already covered in my previous lessons. I have given you a lot of examples about meat analogues, and I have given a lot of examples about the dairy alternatives and so on in my previous lessons. If you can recall I showed you a couple of pictures that I have taken when I was visiting the US supermarkets, and different kinds of analogues or dairy products are already in the market, especially in the US and America. And I also heard from some of you that in India also the market is rising. You can still find a lot of soy milk for sure, may also finding now almond milk and other things. I am not very sure whether you can find meat analogues in our supermarket, but it might come at one fine day very soon. But I know that Indian companies and startups are also working in this direction. So, in this lecture I am going to talk about different types of plant based meats and dairy alternatives, and basically the process we are doing to get them and what are the challenges we have, and those things I am going to discuss. It is not necessarily, I am not going to show how to create an alternative product at this particular lecture. I am going to give you an overview of what is being done today and why we need an analog and why do we need an alternative product. Am I clear? All right. So, again I will come back to my this very second slide from my first lecture. Why do we have to go for alternatives? It clearly shows that animal based products are responsible for greenhouse gas. So, we need to have a transition from animal based to plant based food products, right. And it is clearly shown is that when you talk about whole animal plant based foods, the whole grains, vegetables and all this production or consumption has a very little environmental footprint, and they say that it could be like 20-fold lower, even 100-fold lower than production of, let us say, beef. And that is the reason why we need to have a transition. But I must again have to emphasize the point here is that one of our students were also asking at that time, why do we need to get ingredients? Why can't we eat whole grains? That is always a question we ask in the Indian subcontinent for sure, because traditionally in our cooking culture is that we are less ingredient based, we are more into whole thing based, right. That is the food culture we have, but it is not necessarily the case in every world. And moreover, the world is growing in a rapid pace and we really need different kind of foods, right. We are also following Western food in India also. If you go to Western world, they are also following the Indian food culture. So, now because of easy transportation of people and things, now it is kind of everything is available everywhere. So, we want to produce the food here, but we have to market in the USA. The USA would like to produce food in the USA, they would like to have a market in India. So then basically they have to think about how do we satisfy Indian's tongue, and we should think about how do we satisfy American's tongue, right? And there the food product, the ingredients become super important, and where the processing also becomes super important, so that we can tweak the product so that it gets the desirable taste and texture relevant to that particular portion of people in a particular continent or a country. So I was telling something about meat. I also showed you this figure before. You can clearly see that when compared to Asian people, the Western people, let us say Europe and Northern American people, they consume lot of meat, and the meat is there in their almost daily diet, and the protein they consume majority that comes from dairy and meat in Western world, but in Asian world we get it mostly from dairy and vegetables, right. Vegetable is mostly our main source of proteins, but if in the other part of the world it's still the meat is the major portion of protein, and that's the reason why we are trying to mimic the meat. Just to answer why we want to mimic the meat, because they are not eating meat only for the protein's purpose. It's in their tradition, it's in their culture, it's also in Indian culture. For example, if you have a festival, if you have a marriage, if you have some guests are coming, we bring meat, all right. And it's because it's in our culture, it's not just because we want to get proteins or when we have a guest. No, that's not the idea. We want to have a meat when we have a guest because it's the culture that meaty, that brings people together, right. It's something different, and that's the reason why we need to somehow create plant into meat, right. And that's not easy. Cows can do that one easily, right. They eat grass and become meat. It's a bad way of saying things, but in this case now we don't want to have an animal here in between, because that is having more greenhouse gas emission. Without animals, we need to bring plant into meat. We need to convert insects into meat. How do we do that? And that is where the technology comes into play, right. That is where the ingredients comes into play. That's where the processing comes into play, okay. Now if you look into the market, currently we have plant based, and we have insect based, and we have cultured meat and micro derived alternative proteins. And I was already telling you that the plant based is the major market today. Almost all products sold today is actually plant based, mainly soy based products are available. So it's only the small portion of alternative proteins come from other parts, insect, microalgae and so on. So there is also, I mentioned that there is a huge demand for that one. I don't believe in this number if it is right, because I got a different number from a different paper, but don't worry about the number, but remember that the trend is increasing. We are having around 10 to 15% growth in alternative protein market every year in most of the countries. So when it comes to environment, I also mentioned that alternative protein production has a less greenhouse gas emission, less carbon footprint, plus the biggest thing is that they need less space. For example, to cultivate, to raise a cow for an animal husbandry, you need a large area, but you don't need that much area to grow microalgae or insects, and that's where also you get the benefit. Now we look little bit more into what is exactly needed if you want to convert a plant into meat or milk. Okay, to convert plant into meat or milk, a lot of things comes into play. We just talked about extraction in my previous class, right? Then we talked about analysis, but beyond that we need to get too many things. If you look into the meat analog, just as an example, all right, we discussed about protein. We need a protein with different functional properties, we need a protein with a good water holding capacity and so on, right. But protein itself is not sufficient. We need to have a lot of different characteristics on these meat analogs, then only a human being can consume it, because we have a tongue, remember. So if you just say it's high in protein, people would not buy it. Lot of things has to be considered. The first thing is we need to look for about compositional characteristic. Okay, how much protein is there? People always like it if your food has a lot of protein, what we call halo effect. It's not necessarily true that if you have more protein it is healthy, it's not true, not necessarily true, but it should have a sufficiently good amount of proteins, right. Not only protein, there should be some amount of carbohydrates, not necessarily for the nutritional purpose. Carbohydrates will give you the texture, right. It can acting as a filling agent, it can act as a binding agent, right. So you cannot get the bulkiness of the food without having carbohydrate, and sometimes carbohydrates act also dietary fibers. So there is a lot of advantages, and we also need fat, right, because fat gives a unique flavor when you cook, right. Chicken and fish and all these things, I mean when you cook you get a unique flavor because there is a fat will be oxidized and start releasing small minor volatile compounds, and these volatile compounds gives you taste, right. Sometimes good taste, sometimes bad taste, but often it's a good taste, let's consider it like that, right. And that's the reason why fat is also important. But not only that, fat is also important in terms of diet, because we need to consume polyunsaturated fatty acid. Do you remember PUFA? Omega 3 and omega 6 fatty acids, right. And that should be a part of our diet, because everybody, there are many research articles shows that it's good for your brain, it should good for your cardiovascular health and so on. So you need to consume good amount of fat. And we also need, that's also missing thing here is that we also need minerals, right. We need, for example, we eat meat not only for proteins, meat also contains minerals, right. For example, we need to take a small amount of iron every day, because without iron we can't get hemoglobin in our body, right. We need iron. So if you are continuously eating meat analogues devoid of iron, maybe I not become healthy. Although it contains 25% protein, it is not having iron, maybe there is a problem, right. So we need to create a whole food. Remember, it's not about eating ingredient, we need to create a whole food, and for that we need to add more and more components to the food system. Now this all about nutritional aspect. Then I was telling about even if you have all the nutrients, but if it tastes bad, we will not buy. Your children will not eat food if it is not tasting good, right. Taste is important. Taste is a very generic word here, basically that combines everything, is the flavor perception, is the texture perception, everything should be there. So, and this is the, I would say this is the number one priority in most of the cases, right, taste, right. And we all know that plant proteins are not tasting good, unfortunately. Except soy, most of the plant proteins, even soy has a small flavor, as having this grassy flavor, right. They feel very grassy because they call it as, it's because hexanol or some other kind of volatile compounds associated with these proteins, they come during production, purification and so on. And that grassy flavor, actually people hate that. They don't want to drink milk having grassy flavor. So we need milk that tastes like a milk if you want to consume it. And the second thing is that the color is also important. One of the biggest challenges of plant proteins, we also discussed in one of my other classes, almost all plant proteins are colored. They have brown, they have blackish color, they are greenish color, for example, sunflower proteins. If the protein itself is having a color, it's a big problem. For example, imagine my protein is very green in color, my chicken also becomes green. I don't want to eat green chicken, I don't feel that my chicken or let's say pork, it should be red in color. Meat is kind of a tendency that it should visibly red, then only consumer will buy it. So we can add additional color later during the production and processing and cooking, but we don't want to have our ingredients colored, because that will compromise the final product. And then we need all these other properties which we just discussed, right. It has to have a good structure, it has to have, protein has to maintain the structure, because without structure it cannot maintain functional properties and so on. A lot of work has to be done in this part also. Then we also talk about nutritional value, right. Once you eat, it has to be digested properly. Okay, now it is digested, it has to go to our bloodstream. That should be good bioavailability should be there. And sometime they also come up with some unwanted things. Somebody was talking about, okay, if you look into the package of this plant-based analogs ingredient list, there's numerous ingredients have been added. Whether it's good or bad, nobody knows, right? We try to preserve the color, we try to preserve the taste, that we add ingredients, and that might have a problem, or I don't know whether it's really a problem, but that consumers can hate. And then salt level, that is something we have to be careful, because meat, to make it meaty, you have to add some kind of salt and so on. That might also have a problem. This is something we've already discussed, that the functionality properties are also important. Now, just think how many problems and how many things we need to consider to convert plant into meat or meat analog. It's not an easy task, right? A lot of things has to be taken care to get plant into meat, to convert plant into meat. So, it's not a 1 PhD work, it's a 100 PhD work to get that product. Fortunately, a lot of people in the world in the food science and technology are working in this particular area. So, the field is growing like anything. There's a rapid growth in this alternative food area or analog area at the moment, and hundreds of startups are there and hundreds of companies are finding this is the way forward, because when the government says, because it's not about the science and technology, it's also politically supported at the moment in most of the countries. Most of the country politically it said that you need to reduce consumption of animal based, and automatically the industries are start working on producing more plant based. There's a lot of meat industry, there's a lot of dairy industries have now the new portfolio in their production list plant based. They have been doing meat and milk business like for the last 50-60 years, but now they have a plant based on their agenda. It's simply because we need to have this transition. So, we all need to work together to get plant into meat. Okay? So, how are you producing the plant based products today? We have, let us say, we have a plant cell with proteins, and then we are doing a lot of extraction here, and then we get the protein powder. And the protein powder can be different qualities like 50%, 80%, 90% and so on, and sometimes we need to further purify them, right, another degree of purification, right. This is purification, this is extraction, and once we purified, then we get ingredients, and then we add a lot of other ingredients here. Then you get a food matrix. Okay. Once the food matrix is ready, it is not done. It will be stored, or it will be cooked, or it will be fried, it will be frozen. A lot of things will happen here. At the end of the day, it comes to we consumer. It's again a lot of things involved in this here. We have been discussing only until here in our previous lectures. Am I clear? So, just to give you an overview that the production of single meat analog contains lot of value chains. It's not an easy business. I mean, one person cannot do that, one industry cannot do that. There's a lot of possibility that many people can involve to get the final product that human being can use. Now, I was talking a little bit about converting an ingredient into food matrix, right? Converting an ingredient into food matrix. How do we do that today? Let's take as an example insect, and we have been discussing in our very first class about insect proteins, and when I asked you the question, how many of you would like to eat insects, nobody raised your hand, right? Some part of the world they do eat, but the rest of the world they don't want to eat insect as a whole food, simply because there is a culture, there is a perception that it's not good, oh it's a bug, oh it's carrying some disease, I don't want to eat it, right. If I say, can you eat house flies, can you eat cockroaches, but they are also 50% proteins I am talking about, right, but we don't want to eat them whole. However, to solve this problem, then we have all these technologies, convert insect into a protein powder, right? But can we eat the powder directly? Can we eat wheat flour like this one? No, we make chapati out of it, right? We need to make bread out of it, and then only we can eat. Unfortunately, we can't make chapati and bread directly out of plant proteins or insect proteins. That's also a problem. Wheat flour has a very nice gluten network and make nice chapatis, nice rotis and all these things, right? But insect proteins, I don't know, the roti will have this, paratha will not have that stretching behavior. You can't make that dough. You can use it as a 50% replacing wheat flour, but not 100%. Then there is a problem again, right? Then we need to do again processing. One of the processing we are doing today is extrusion. Have you heard about this word? I am going to give an exclusive lecture on extrusion later in next half an hour, where we convert this powder into a new texture, because if you want to have a chicken, it has to be fibrous in nature. If you want to have a beef meat, it has to be like juicy, that texture should be there. If you want to have an egg, you know that egg, that grainy kind of thing, that texture it has to get. So to get all these textures, lot of processing can be done. One of them will be extrusion, but you can also do a lot of this one, homogenization, texturization, many other techniques are there. So again you need to process it, and then you get, let's say, meat analog or maybe milk as an example. Now what we are doing exactly, just by processing everything, instead of eating insect directly, we are eating the insect products. Can you see? So this is where the technology is heading to nowadays. You don't have to eat insects directly. And it's also not possible to eat microalgae directly. We talked about a lot of problems here, because microalgae is not digestible in our body, especially let's say chlorella. It has a very strong cell wall. Our stomach does not have cellulase enzyme. You can't digest it completely. So, however, if somebody can get the microalgal proteins, maybe we can make a meat analogs out of it, then we can consume it. Okay? Now, another thing is that, especially talking about processing. So, if you look into this one, a plant based thing has so many ingredients. Can you see here? Right? It has binding agent, it has antioxidant, it has minerals, and it has acetic acid. All these things are added. They are not naturally present. Keep that in mind. In the natural meat, the animal has created in the biosynthetic pathway, right? That's all natural. But in case of artificial meat, we take proteins from soy, right? We take vitamins from plants, okay? We take minerals from some seawater, let's say. We take other things from calcium from somewhere else, right? Everything is sourced from different sources, and they're all mixed together, and then you get the final product. And in between, what do we have? Processing. And this is a big question mark at the moment, whether we need to do processing, whether it's good or bad. Without processing, you don't get the texture, you don't get the color, you don't get the flavor. But processing also brings some problems sometimes, right? Have you heard about the word ultra processing? Nowadays, there is a big debate is going on around the world whether we should use the word ultra processing or not, right? Because when you write the word ultra processed, consumers say, oh, maybe it's something super bad, I don't want to touch it, ultra processed. They say that maybe bread is also ultra processed, if I'm not wrong. UHT milk may be ultra processed. It's more about how much heat load, how much processing and other things it has. But without processing, we don't get anything. But there is a big question mark in this area, how the processing and ingredients are affecting the final quality of the food product. Are they creating some unwanted things? Are they creating things which we don't know? Because we need to feed this to our young children, we need to feed this to our elderly people. Can they metabolize all these things properly? Is there a problem? And there is a huge research gap in this particular area. So, I recommend that if you would like to pursue research in this area, you can think about processing induced modification of proteins, processing induced changes in food, how that affects human body. There is a huge research gap. Nobody knows, because we are using top down approach today. What do you mean by top down approach? That means we first create the product, then we give to the human being, right? Then we wait whether the human being is happy or he will die. We never know, because the human dies slowly, you don't die immediately, because you are not feeding poisons, right? right? But it has to be bottom up approach, because we slowly need to understand which ingredient is bad, what is the problem with soy, what is the problem with added minerals, what is the problem with added emulsifier, that we do not know, right? Everybody is talking about taste, flavor, texture. If it is tasting good, if the flavor is good, if the texture is good, okay, let us get into the market. That is what exactly happening today. It's not, I am not telling it's bad, because we need to have some transition, it's good. However, how that is affecting the human body in the long run, because we want to keep the human race for several years, several generations. Our food is actually defining how we behave. Our food defines how we produce our offspring. How our food defines how we think, right? All this linked with the food. The question mark is, okay, whether or not these plant-based analogs or meat are good or bad. I do not have any data on that one. I am not telling that it's bad. Please don't take that as a take-home message. We don't know, and there's a lot of room for research in this particular area. Okay. And now I'll give you a little bit examples on some of our research, what we have been doing in our university to get some of the analogs, some of the alternatives. We do a lot of research on fermentation. I'll talk about fermentation exclusively in one of my future lectures. So today I don't have much time, I have to delete all the slides, but I'll take it in my future lecture. So we do fermentation, because if I was mentioning that this processing what we do, heating, cooking, boiling, extrusion, everybody thinks, yeah, it might be good or it might not be bad. But we have been using historically fermentation, right? Some of our foods are also fermented. In India also, China, soy and tempeh in Indonesia, they are all fermented foods. So we have a strong tradition in the modern world, fermentation, and we believe that fermentation is good. So in this work, one of our colleagues, I think, I don't recall her name now, she did a lot of nice work, and she could convert this pea protein isolate into cheese. Can you see? Cheese blocks. And we have a guest lecture on the last day, on the 16th of October, where one of my colleagues will talk exclusively about how plant-based proteins can be used to create a cheesy structure. So I don't take the case studies today, okay? So that will be taken in the next exclusive lecture on the last day, okay? Then we also have another colleague, Rene and others, and they have been working on mushroom. Mushroom is a fungi, right? And it also has a good amount of protein, and those mushroom mycelium can be fermented or can create a very meaty texture. They did took a fermentation approach, and what they found was that this fermented hyphae, after heating, it gets kind of a meaty flavor and meaty texture. So basically why I am highlighting fermentation here is that there is no much processing here. There is a processing, but that processing is done by bacteria, right? A gentle processing can be used to create a food mycelium, a fungal mycelium into meat analog. It is a huge research on fermentation to create meat analogs. And one of the things I wanted to mention is that about consumer preference. As I mentioned, that everybody thinks taste, texture, appearance is very important. Without that, they won't buy the alternatives. Now let's take a meat as an example. It's such a beautiful thing, meat, right? Not good for the environment, but to look, it's such a beautiful thing. It is so nicely structured. For example, it has this nice red proteins, right? And then you have a thin layer of fat, and then the protein, then fat. There's a lot of interfaces here, and there's a lot of water holding proteins inside, and that gives you the juiciness, right? And meat is not just meat. In some part of the world, we like eating only chicken breast piece, but if you come to India, we need bones, right? We don't like to eat only chicken breast piece. We really want to eat chicken attached to the bone and tear it off, and then it is, right? Our perception is different. So we don't want to have eat soya chunks. We want that feeling. We need bones, right? And there's a lot of things has to be done. How do we get that entire thing from the analogs? It's a big challenge. I have no idea how do we get bones out of plants now. Okay? To get this texture, right? To get this shape, now meat is like, see how beautiful it is. We have a spider here. We need to get the shape of the food, and for that we need some kind of designs, right? Now we have a new technology in research and development. It's not in commercially available, which we call 3D printing, right? You must have seen 3D printing. It's used in industry a lot. To 3D print, you can 3D print anything today. You can 3D print human beings shapes. You can 3D print plastics based things. You can 3D print a computer design, anything can be blocks, everything can be 3D printed. Basically, you tell the computer which design you want in a 3-dimensional way, and there will be an ink. The ink could be plastic, ink could be anything, and it will be molten, it will make the design what you want. Now we are using 3D printing in the food science also. Instead of the ink, we are going to eat food ingredients, right? You put food ingredients in the inkjet, and now they will print the food you want in the texture. Oh, I deleted those slides at the interest of time, but I'll also take 3D printing in my next classes. So we can create different shapes of food, because children don't like every shapes, right? They like, for example, I have my sister's son, and if I take him to KFC, he doesn't like the big, that roasted leg piece. He likes that small, what is it called? Popcorn chicken, right? Nuggets, exactly. He likes nuggets, right? So how to get that nugget shape? We can get the 3D printing, right? With the plant protein isolates, we can get a nugget structure. There's a lot of things going on today in the market. As I mentioned, just to showcase, things are already in the market, but more work is needed. Why this more work is needed? The first thing is that inadequate protein intake. That's one of the challenges we have today in the market. The second thing is iron deficiency, because the meat analogs might not necessarily contain a good amount of iron, right? This is because iron present in meat and plants in different forms. Doctor says that if you just take iron capsules, it might not be bioavailable completely. You heard about the bioavailability, right? Every minerals is not available in the same form. They would like to be in different forms. Some are attached to some organic molecules, some are attached to something else, some are attached to proteins, and then they are bioavailable. The free iron not necessarily but not bioavailable. So even if the meat analogs are added with lot of iron, those irons might not be bioavailable compared to the iron present in chicken liver, for example. Doctor says that if you have iron deficiency, eat some livers. I don't know whether it's good or bad, but they eat that one, right? Because they say it has a good amount of bioavailable iron. So by mixing things, we need not get the right bioavailability. Although on the paper it says 1% of iron, but maybe 1% of iron may not be equivalent to 1% of iron present in real meat. Am I clear? So those challenges has to be addressed. Third thing is vitamin B12 problem. Dairy people, especially vegans, we have a lot of dairy analogs today. Vegan cheese is there, vegan milk is there, vegan egg is there, vegan chicken is there. Also a lot of other analogs coming for this one. But dairy is one of the good sources of vitamin B12, right? It's an important, present in a very small amount, but it's very important vitamin for our body. And if you don't consume it, you will really have a problem in health and diseases. Metabolism can be compromised, and we don't get if you completely turn into plant-based or vegan diet. How do we improve the vitamin B12 content in plant-based foods? A huge research is going on this particular area. Certain bacteria can produce vitamin B12. Certain other things can produce vitamin B12. Then they are thinking about, okay, can we ferment with this particular bacteria, or can you spike vitamin B12 onto food matrices? There is also research scope in this particular area. And another problem is that plant also lacks calcium, meat analogs. So milk contains lot of calcium. Calcium is important for your bone development, right? And calcium has to be present in the right form to be bioavailable, right? Calcium has to be present with vitamin D to make its bioavailable. There is a lot of combination because food is a complex mixture. They are present in a combination, then they become bioavailable. It is not about the number game. It is not about always quantity in food. It is more about quality, digestibility, bioavailability, and so on. Does that make sense? And I did not talk about the anti-nutrients yet. We have an exclusive lecture on anti-nutrients in the coming class. Plant-based analogs, meat-based analogs, plant-based meals can also come with lot of anti-nutrients. Nobody knows how it is actually, whether it is a problem or to what extent it is a problem. We know little about trypsin inhibitor, phytic acid, lignans, oligosaccharides, saponins. Only this much knowledge we have at the moment, but how they are affecting the body, we don't know yet. Okay. And I mentioned that at the end of the day are all processed. Nobody knows how processing affects. So these are the key challenges we have today on the plant-based processing world. Does that make sense? So this is what I wanted to tell you about where are we today with respect to meat analogs and dairy analogs. We are more focusing on the texture, flavor, taste and the quantity, and there is a less focus on digestibility, health effects, anti-nutrients and so on, and those things has to be balanced to make the plant-based foods or the alternative foods sustainable. Not only sustainable, it is also good for the human being. Please, health is a part of sustainability. If you save the world, but if you die quickly, that is not sustainable, right? It has to go hand in hand, right? The world is for us, not only for us, but we are the part of us, right? And there we are at the moment. I am just highlighting you where are the research gaps we have that you can take in the future if you want to take a plant-based research in the future. So we will take a break now. So before I go to the extrusion lecture, the last lecture of today, let's take 10 minutes break and I'll give you a short lecture on meat analog preparation based on extrusion. Is it okay? Then I'll give you. Don't worry, I will not take a long lecture, a short one. Is it okay? 10 minutes break. We'll be back. Welcome back and welcome to the last lecture of today, the lecture number 9, texturization techniques, high moisture extrusion and shear cell technology. So in the last lecture we have been discussing about we need to create plant based meat analogs or alternative protein based meat analogs. It's good to do that to get the desired texture, flavor and color and so on. It's also very important. However, how do we get that particular texture? How do we get that meaty texture? How do we get that fibrous texture? How do we get that juiciness? For that we again have to process our protein ingredients together with other components. It could be water, it could be fat, it could be carbohydrate, it could be some other binders. So everything has to be mixed and reprocessed to get the texture, the meatiness, the fibrous nature of meat that we want. How to get that one? We need to again have to process. And one of the most common processing technique which is industrially employed, it's not in the laboratory scale I am talking about, not only in the laboratory scale, industrially employed is high moisture extrusion. Okay. I am going to talk a little bit about high moisture extrusion. Just recall my slide again. We had insect, we did not want to insect, I convert into insect protein isolate. Now by extrusion I can get different texture or fibrous nature I want, and that can be used as a meat analog, and that is where we can have insect indirectly. Okay. Have you done extrusion before, anybody here? You have done it? Maybe I personally haven't done extrusion myself, only my colleagues have done it for me. I analyzed you have also done it. But let us see that you are also welcome to share your experience if there is a question. Okay. Extrusion is a big machine, in other words, right, where things are extruded, in other words, right. This is the basic structure of an extruder. What does it have? It has a feeding chamber, right, where you introduce your protein as an isolate or a concentrate. Of course, you are also welcome to add other ingredients together with it. It could be carbohydrate, it could be a fat, and everything will be added to here. And then basically it comes to the second region, what we call kneading zone. Is kneading is nothing but a mixing or doing something like this, right. In that particular zone it comes where all the ingredients that you have added will be mixed with water. Okay. Water, you remember, water is important for what? Hydration, right. To improve the water holding capacity, we need to introduce water, right. It is called as high moisture extrusion because the water content can go up to 80% here, like if I am not wrong, people call it as 60 to 80% will be the water content in the product. That is the reason it's called high moisture extrusion. There is also something called low moisture extrusion, where the water content will be like 10 to 40%. Okay. I am not going to teach that today, because that is not very often used for the juiciness, right. So, we use high moisture extrusion, the water content in the product will be 60 to 80%. So, everything will be mixed here, and then the product will be sent in this direction. We have a twin screw like this. You must have seen it in mechanical engineering laboratory, how that exactly the twin screw looks like, right. It is rotating like this one, and while it is rotating the screw, it will take all the ingredients together with, and it will be mixed at very high shear stress and pressure and also temperature. The temperature in this particular region, in these regions, can be between 50 to 180 degrees Celsius. Basically, you are cooking under high pressure, right. You are cooking the plant proteins together with ingredient in a screw at high temperature under shear stress and pressure, and that will actually change the protein structure. Now, the protein, it was originally looking like a powder, it becomes fibrous, right. And then it comes to the end of this one, there is a cooling zone where basically the temperature can go like 50, 40, something like that one, which were very, very hot, will be immediately cooled down, and then extra moisture will be evaporated there because of the condensation here, and then you get the product out, which is exactly looking like a meat analogs. But you can again have to cook it, you can again have to freeze it, you can again have to thaw it, you will also do other processing after getting. The one you get, it is not necessarily the final product, they still process it. And this is the basic principle of extrusion. Is that clear? It is one of the most commonly used method today to get the meaty, juicy, fibrous structure of meat equivalent to meat from plant proteins or insect proteins and so on. Make sense? Very simple. There is a big machine. You have a feed mixed with proteins and other ingredients that you like. They add also colorant, they also add some other things you want, and that will be sent in this direction, and then it is mixed with water, right, and then it will be moved in a twin screw at high pressure, shear force and high temperature, where things will be cooked. Then the protein denaturation happens, and that will lead to the fibrous structure when you cool it down at the end. That can be further processed, like you can do boiling, cooking, freezing, thawing, many things they do later to get more fibrous structure. Make sense? So, now the most important thing for us to know, why do we get that fibrous structure, how and why, what is the mechanism here. There is nothing has been proven it actually, but there is a lot of hypothesis, there is some basic experimental studies. They postulate that these things can happen inside this machine over here. Okay, over here many chemical reaction can happen, that is what they are trying to say here. The first one is that in this particular zone here, you mix it with water, right. In this particular zone here, you mix it with water, and the temperature can be like 40, 50, 60 degrees Celsius there, right. And some proteins will have a denaturation temperature 50, but some will be usually having 80, 90, 100 and something like that. But now the protein is in contact with water, and now it is actually mixed. Coming back to our first lecture today, solubility and water holding capacity, right. Now protein is coming in contact with water molecule, the protein will be hydrated, right. Water holding capacity of the protein is increasing simply because now more and more protein is in contact with more and more water molecule, right. And then in this particular zone, in this particular zone, the temperature can be, they say 100 degrees Celsius to 180 degrees Celsius depending on the machine here, and that is the temperature. We all know that most of the plant proteins can also denature, right. Globular proteins usually have a very high, the albumins usually have a high melting points, melting temperature, but at 180 degrees Celsius I can tell you that most of the proteins can be denatured. When the protein denatures, it will expose all its functional groups, right. It will expose all your functional group, and now that protein is available for several chemical reactions. Number one, it can have a protein-protein interaction, it can have protein-carbohydrate interaction, it can undergo protein oxidation, it can undergo protein-lipid interaction, because now it is exposed and everything is mixed together in one zone now. There is a huge nice contact between the molecules, and there is a lot of opportunity for chemical reactions, right. And then that will also help because you are screwing in this way, and it helps in the stretching of the protein. At the end of the day, you cool it down. Then the open proteins, now it has undergo chemical reaction, or it can actually form these cross-linked networks, right. Now we started with the globular proteins, right. Now you got a network, can you see? You started with the individual globular protein like this. Now they are actually a cross-linked network, and that cross-linked network will give you the fibrous structure according to the literature. I am not very sure it is 100% true this is a chemical reaction, but most of the people believe that this is the way you get the structure, but there is a lot of other phenomena as well. Does that make sense? So you just converted a globular protein into a fibrous protein now, because of the chemical cross-linking. I just talk little bit about chemical cross-linking, just you no need to read this again, I will just show you. So the protein will have 3-dimensional structure like this one, and there will be intramolecular disulfide bonds, and there will be intermolecular disulfide bond between two proteins, right. When you heat it at 180 degrees Celsius ish, right, there is a possibility that these bonds can be broken because they can undergo oxidative modification, reductive modification and so on. Then basically you will break this intramolecular disulfide bond, then the protein will be exposed like this one. Now when there is a thiol group is exposed, and if there is another protein with a disulfide bond like this one, they can interact with each other and can form a new cross-linking like this one. So you are creating a protein-protein cross-linking, and the protein-protein cross-linking can lead to the aggregated proteins, and that can essentially give you the fibrous structure. Does that make sense? The take-home message is that in the different zones in the extruder, opening of the protein takes place, chemical reaction can take place, protein-protein interaction can take place, protein can be covalently cross-linked, and that will give you the fibrous structure. Any question here? Is it clear? Yeah.
Sir, in case of low moisture content, this extrusion that is working on low moisture content, the dye from which this compressed material is coming out is having major role for the product development.
Okay.
So is there any relation or correlation between dye and high extruded product in high moisture content?
You mean the dye, this particular portion you're telling here?
Yes, sir. Sir, in case of low moisture extrusion, it's having a big role.
Yes, of course.
Because
yeah.
of the high pressure and low pressure. Outside, inside it
yeah,
is,
from here to this transition
it's
point.
in high
yeah.
pressure. When it comes to low pressure, it expands. So that makes the product
Swelling,
fluffy, and its texture is developed in from that dye.
Yeah.
So is there any relevant effect on in?
Yes, I think one of the relevant effects I read. I honestly tell you that I never did this experiment myself. What I read was that one of the most common thing happens when the sample comes from this region to this region. They expel the water which was not actually holding on the proteins, basically kind of a syneresis, like it loses the water, and apparently that might have an effect on stabilizing the structure, because now you don't have a liquid kind of a jelly matrix, but you will get a matrix which is juicy, not like a yogurt. That is the one thing I know, but I am not very sure how exactly the pressure change will have an effect on final product. I have no idea, unfortunately. But what I know that they claim that when the things come from this region to this region, this is what happens. See, the cooling. This is what happening at the dye region. They say that the stretched one, now they start cross-linking when it is cooled. I'm not very sure whether it's true chemical reaction that's happening. I don't see any very strong chemical basis for that one. But this is what people believe. I don't see a very strong evidence for this ever. Yeah.
Sir, one more point.
But do you think something will change? Because you have experience, I don't have experience.
Sir, in case of low moisture extrusion, the water is having big role.
Yeah.
So when it is compressed under high pressure and high temperature, when it comes out of the dye, it expands.
Yes.
So when it expands along with the product. So
exactly.
we get cells there.
Yeah.
We get the particular shape, and it depends upon the shape of the dye. Which type of product we want. We want any customized product, we can change the dye. Similarly, I do not have worked on this high moisture extrusion, so I just want.
I'm not very sure. Sorry, I don't know how exactly the dye, its shape and its temperature have an effect. I don't know. Let's find it later.
Okay, sir. Thank you. Thank you, sir.
Good. This is how the commercial scale extrusion equipment look like. This one, they come in different sizes. Huge ones are also available. We can also buy a laboratory scale of this size. Thermoscientific sells one laboratory scale extruder that can do laboratory scale experiments. But they're also available by different companies in a very big scales. Okay. This is not something new, right? Extrusion is not something new. You know pasta making? Pasta, you get a shape, and in chakuli, what we call in English? Chakli is, I don't know what is the equivalent in Hindi. Chakli, same. Okay, they're all basically all this. It's a similar phenomenon, right? Muruk, whatever it is. Like it's basically, we don't do it with extremely high shear force there, but still we apply a shear pressure, right? It's basically 3D printing according to me, making chakuli and making all these things, all 3D prints. Now it's already there in our culture, right? But now we have machines to do that. There is another story, right? And also the noodles and all these things, they're all actually, but extrusion has a little bit high temperature and that twin screw, all these things are there. But otherwise they all follow a similar thing. For your understanding, I'm telling, it's not something completely new, okay? And you can just see for the new people here, you can just see that how different kind of proteins produces different kinds of texture. It's a nice figure, I found it from this review article. You can see that it has this chickeny structure. Can you see? Usually wheat gluten has these disulfide bonds and sulfur atoms, thiol groups that help you to get this stretching. In bread making, the dough gives you that stretching, and that can also be exploited here to get the meaty fibrous structure. It looks like a chicken breast piece. Can you see? It looks also like a chicken, but I do not think this is looking like a chicken though. And I have two other things to example here, like the problem with the color, remember. Now if my chicken is having this color, we make a green chicken curry in our hometown, maybe you also make it here, but we add palak or something later, but we do not want a chicken which is already green, right? Consumer's problem. So if the protein is green, we have also problematic here, right? So that's the reason why you need to find a right ingredient. You need to remove the color from the protein to get a real this color, okay? Now I will show you little bit about some concern. One of the biggest thing we have to remember here is that extrusion happens at high shear pressure or force, whatever it is, and the temperature, the heating can go up to 180 degrees Celsius, right. It's good, it's good for cooking, I fully agree with that. However, high temperature can also lead to some of the unwanted chemical reactions, right. High temperature can also lead to some of the unwanted chemical reaction, because you have protein there, we have lipid there, you have minerals there, and they can lead to some unwanted chemical reaction, and they can form new type of chemical compounds, and nobody knows how that will affect your body. And I have received around 6.5 million Danish kroner to investigate this from the Danish, around 7 crore Indian rupees, to do a research on how exactly the chemical changes can affect the digestibility, and we are also working on how it will affect the brain. If you continuously eat extruded product, whether you will sleep more in the classroom. We never know, right, the digestibility, right. So those things we are going to, not sleeping in the classroom, we are investigating, but how it affects the memory. You learn so many things you might forget because of eating lot of extruded product, I do not know, we are going to investigate that one. Now I will go to the last part of today's lecture. Similar to high moisture extruder, we have another technology called shear cell technology. It's kind of somewhat new, I would say, not available everywhere. I did not see a lot of articles on this one. It does a very similar job like a high moisture extrusion. It also creates an anisotropic fibrous structures using a different mechanical phenomenon. In there you have this twin screw. Here you basically have what we call shear cell, okay. I also personally haven't seen this machine and never used myself, but I am only teaching you based on what I have read here. Apparently it has two cones, a cup, a conical cup here, and it has another cup here on top of that one, and the sample is placed. I hope I have a diagram here exactly. So you have a cone here, the first cone, and we have another cone on top of that one, the second cone. Can you see this part, right? And your sample, the protein, carbohydrate, everything is placed in the middle. The lower cone, apparently it rotates, and then it creates a very high shear force, and this temperature can also be controlled here, okay. You can apply high temperature, and that apparently gives you a very high fibrous structure that can be used to create meat analogs. It's a new technology. I'm not a mechanical engineer, unfortunately I can't teach you so much about the machines. Maybe you guys are cleverer than me, you have a bachelor's in technology, but I have a bachelor's in science. So basically it has a two cups, two cones, and between two cones the sample is placed, and one of the cones rotates, and that creates the shear stress, and that leads to the fibrous structure. And the temperature is also important here. It also goes to high temperature. Apparently you can control the temperature here. Mechanism exactly the same that I just explained: protein denaturation, renaturation, and cross-linking. You are converting a globular protein into a fibrous structure, okay. If you really want to know this shear cell technology and how to create the fibrous structure using protein, I really recommend you go through this video. Unfortunately, I can't append the video because of the Gian guidelines. I have put a link for you here. It's a 1 hour 13 minutes video, but it has a lot of information about how to create a structure, meat analog structure using plant proteins with different technologies and what are the parameters play a role here. It's explained very detailed in here. Okay? I really recommend that you go through it when you are free. Then you can watch a YouTube video, movies and other things later. All right, and that is the class for today. So, we discussed about several things today. I really cannot summarize from the morning so many things we have discussed, but in the afternoon in this current session we discussed about two important topics. How do we create meat analogs and dairy analogs, and I talked mostly about some of the challenges. I told you about it's not just proteins, it's too many other things are involved to get the final product. It's never a just protein. My lecture is about alternative proteins, but only protein alone cannot work. It has to combine with so many things to get the final food product. To get that, you need to do a lot of processing, you need to add a lot of ingredients. They have a good and bad effects. A lot of research is needed in that area. Then we talked about two important technologies that we use today, high moisture extrusion and possibly in the future the shear cell technologies to get the meaty fibrous structure. We realized about the chemistry behind that one. It's protein denaturation and cross-linking, apparently that leads to the fibrous structure, but there is a lot of other physicochemical phenomenon that we do not have to worry about. But if you are really interested more about getting the texture, go through this particular video. And that is all for today. Thank you so much for attention, and let us see you guys tomorrow. We are going to talk more about digestibility, bioavailability and so on. Thank you and enjoy your afternoon.